I want to welcome you to London, to Imperial College, and to the UK Dementia Research Institute. This is, Henrik and I, are really excited about this occasion. Many of you have also traveled an awfully long way. There was one casualty of travel. Unfortunately, fortunately, he's safe. But Nico Barthelme didn't get off the tarmac at St. Louis. I will try to show later a rather remarkable photograph of two airplanes shaking wings on the ground. So we hope that Nico will be able to join us by Skype or webinar later today. No one was hurt, but it was he spent some unpleasant hours in the airport. But in addition to thanking all of the speakers, I want to thank all of you who've come, because it's really important to recognize we've intentionally kept this very small and tried to constrain the meeting in a room that was of human size in order to encourage discussion. So the hope is that everyone is going to feel as much a part of the meeting and as much of a contributor to it as the lecturers themselves. This is a workshop, and I was trying to think of where the word, how the word workshop developed. But I finally, as I was reflecting on this, I realized what we've done here is we've invited some expert craftsmen to work with those of us who are apprentices or setting up our own shops or merely just interested in the product. So we're all in this together. Now, as we go through the day, I hope that everyone can not only absorb new information, but also start challenging the current state of the knowledge and asking really fundamental questions, at least as they see it. And the sorts of questions that Henrik and I were thinking about, as we put this together, are, questions like, what factors determine NFL concentrations and their distributions between blood and cerebral spinal fluid compartments? And then how do these concentrations, how do the pharmacokinetics of this molecule relate to the underlying neurobiology of the intermediate filaments? These may help us, I hope, by the end of the day, focus down. I have a rather translational focus in this, but focus down on what we really need to know about intermediate filaments and neurofilaments in particular in order to develop confident clinical interpretations of changes in the levels of these molecules in the different tissue and cell compartments. And that kind of gets back to some really interesting emerging neurobiology about how these proteins are processed in the neuron, what the mechanisms of release of proteins from the intracellular compartment are, and of course also on the mechanisms of large molecule transport between cerebral spinal fluid, brain parenchyma, and blood. The hope is that in thinking through these questions and in trying to crystallize some of the key questions as we go through the day, we might be able to end up the day with actually some sense as a group of what key experiments might be. Because we have some representatives related to funders, some of those in industry here who really are trying to use these markers to develop better drugs. The hope is that we can, as a community, help direct ourselves and then help identify for those people who are managing research portfolios what we as a community need to do. Well, so I think that's enough for me, and I'm going to close and pass you on to Henrik for some opening remarks in a moment before starting. But I just wanted to say before that a couple of very important details. First, as you can see, we have people filming. All of the lectures will be filmed today except one, where there's going to be some privileged information communicated, which is very exciting for all of us who are here. I just want to make sure that everyone in the audience knows that it's being filmed, so try to keep the ambient level of noise down. And if you're uncomfortable about having your words caught on film, and you do want to say something, please let us know, and just by raising your hand or something, and we can shut off the video if you have something very more confidential to say. I hope otherwise that people will be happy being videoed. The intent is to share this video on the website to allow many people who couldn't join us today to participate as well. So I hope this is really a benefit for the community. And finally, not least important, I want to give a big thank you to the funders for this meeting. They've been terrific supporters. First, I want to thank Biogen and Devangi Mehta in particular for really quite remarkable support of this, this concept, the idea of an open ideas exchange. Biogen had no role whatsoever in identifying the speakers, in planning the meeting, in guiding Henrik and I to what we were doing. In fact, they took pains to step back. They just wanted to see the science, to learn about the science with us. And so I think we owe them a great thanks. And I also want to thank the UK Dementia Research Institute, which is a really quite remarkable research effort kicked off by £250 million of government and charity investment over the last few years. It's a research institute that stretches over 6 major universities in the UK, UCL, Imperial, King's, Cambridge, Cardiff, and Edinburgh. Henrik is a professor in the UK DRI at UCL, joint with his appointment in Gothenburg, excuse me, and I head the center here at Imperial. So this is an institute that we hope will foster many more such exchanges as this and catalyze new opportunities for research here and abroad through a common interest in solving the problems of dementia. So let me stop there and pass on to Henrik for additional comments.
Thank you. I will be very brief because you put it so well. This was exactly how Paul approached me about this day. And I felt this is such a great idea. Let's do it. So, and now when we look at what will happen today, I think it's very timely and perfect. And we need these types of discussions and talks from basic biology to the biomarker side. And the first session will be really a mix of this. And I am very thrilled about this day because I think we will learn a lot. And at least I need to learn more about the basic biology. Neurofilament, in my home university, it's not a big university, it's also Sahlgrenska University Hospital. There was a neurologist, he's still alive and working a little bit. His name is Lars Rosengren, and he spent his PhD studies on neurofilaments. So he, in the 70s and 80s, he purified neurofilament heavy, light, and medium from bovine brains and also macaque brains. And then he made the first antisera. And then he actually made an assay that worked so surprisingly well, and that was the first assay we started to use in clinical neurochemistry routine, and that was the assay that formed the basis of the first commercial assay that Human Diagnostics made up in northern Sweden, really leaving Lars Rosengren completely without any recognition of that. I think he's rather unknown actually, Lars Rosengren. But now there are more assays coming along. We also have these possibilities starting to measure the marker in blood, neurofilament light that is, neurofilament heavy and medium, there are also assays. And it's so from a biomarker perspective, a lot of things have happened. And there is so interesting biology going on around the neurofilaments also. And I think we need to learn more about that biology to, you said that, Paul, to interpret the biomarker results. So I think I will I think I start by introducing the first speaker, Jean-Pierre from Laval University, Quebec.
Okay, thank you very much. Okay, I need to change that.
Might want to use it. Might be even better now that I think of it.
Okay.
See, are these live? Let's see.
Need to change the.
Do I have to do anything to make this? Oh, there we are. Perfect. Perfect. Good. So
And then I do this.
there you are, Jean-Pierre, if you want to.
I need to change the presentation.
So, it used to be that I would drop the slides before coming up. I mean, those of you who are younger won't remember this, but we used to carry around acetate slides, and they were really awful because you had to plan weeks ahead of time, and then you'd walk up to the podium, and they were carried in these carousels. And if you didn't put the top on the carousel when you handed it to the person who was about to put them on the slide projector, they just all dropped out on the floor. But Jean-Pierre.
Yeah, it's much better with the PowerPoint. First of all, thank you very much, Paul and Henrik, for inviting me. I think it should be a very interesting meeting. And it's always fun for me to come back to London also, because I did my postdoctoral in London at Mill Hill. I think they destroyed it now, but in the beginning of the 80s, that was one of the best places in the world to clone genes, and I will come back to this later on. I have a disclosure. I'm involved with a small company, small biotech, in development of therapeutics for ALS, and I will discuss at the end one of the compounds we're working with. Neurofilament, I became interested in neurofilament as a PhD student. I was with Walter Mushinski at McGill University, Department of Biochemistry, and my project was to work on synaptosome. I was doing gradient centrifugation and took electron micrograph of all this, and it turns out that I was taking electron micrograph of neurofilaments. They were contaminating my preparation, and Walter told me, why don't you work on neurofilament rather than synaptosome? It was so bad to prepare synaptosome. This is how it started. And the structure was not known at that time, but now we know from the DNA sequencing what is the structure. The main neurofilament protein in the adult nervous systems are neurofilament, NFL, NFM, and FH, and alpha internexin in the CNS and peripherin in the peripheral nervous system. And neurofilament, they have also long projection tail domain that we know they are highly phosphorylated, lysine, serine, protein repeats, and that wasn't known in those days. But we knew that the filaments had kind of projection domain on the structure. And I found very early on how to isolate these domains. You just treat the filament with chymotrypsin. And if you do a centrifugation, you will have in the pellet a fragment of about 40 kilodalton that represent the alpha helix rich domain. We know now that. We used to call them binding domain. And in the supernatant, you end up with one major band there. It was from the neurofilament and FH. In those days, we call it 200, 145, 68 for H, L, M, and H. And all three protein had a 40 kilodalton after digestion with chymotrypsin. And with antibodies polyclonal to NFH of the different protein, we're able to find that in what was released in supernatant was a projection domain of NFH. And they were highly phosphorylated. When we prepared the phosphorylated neurofilament following injection of p32 octophosphate in the rat brain, we would find that all the phosphate, majority of phosphate, were in the tail domain of NFH. So this structure that we showed in those days for NFH and NFM resemble what we got after that. We didn't know at that time where was the C-terminal and N-terminal, but that was basically the basic structure. We knew also that they were highly phosphorylated. This is a JBC paper we got in 1982. This paper was rejected from Science because the editor said that it's impossible. They have too many phosphate per whole protein. We don't believe it. And we ended up with 22 mole of phosphate for the NFH, 9 mole for NFM, and 3 for NFL. And if you treat the filament with alkaline phosphatase, there was a shift in apparent molecular weight of NFH, showing that there must have been a lot of phosphate. And on 2D gel, there was a shift in the isoelectric focusing, so further demonstrating that they were highly phosphorylated. But we didn't know much more about this until we got the sequence of the genes. And this is what I did when I came to London. I have cloned neurofilament genes. First the human NFL and the mouse NFH, and other labs, Bob Lazzarini in the state at NIH did the human NFH, and Nick Cowan in New York about that time did the mouse NFL. So all this came out at about the same time. And this is the introns exon sequence of human NFL. Actually, it took me almost 3 years of postdoc to clone neurofilament genes and sequence 12 kilobase. So I think nowadays it would take me 1 day to do a postdoctoral. So that was tough in those days. But the sequence of NFH was very interesting. It revealed the repeat sequence, KSP, KSP, KSP. In the mouse, there was a 53 or 48, depending which strain of mice that you may end up with different, but over 50 KSP repeats, which was more than what we've determined by a number of phosphates. So the potential for a very high number of phosphate in NFH tail. So the availability of neurofilament genes allowed us to generate transgenic mice. And this is what I did. First thing I did when I started my lab in Montreal is collaborate with Alan Peterson at McGill, and we generated transgenic mice expressing the human NFL. So it was the intact gene we put in the mice. And the genes, the transgene was expressed like the endogenous. We had an antibody from Denise Paulin from Paris. She had an antibody that recognized only human, not the mouse. So we could use it as a marker, and we get very nice staining here of human, not in normal mouse. These are spinal cord. And the mice were pretty normal, except some cells showed the accumulation in the cell body of NFL in the brain, especially in the thalamus. And you can see some of human NFL there in motor neuron. But nothing much in terms of phenotype in the mice. The big phenotype came in one paper published in 1993 when we overexpressed human NFH in the mouse. It was the normal human NFH, 44 repeats of KSP. In human, there's either 44 or 45 repeats. The 45 allele is slightly more frequent than the lower allele. And I'll come back on this later. And this created, overexpressing NFH created large accumulation of filaments, you could see by electron microscopy, in the cell body of motor neuron. And it created an atrophy of the axons in the ventral root. You can see a Bell5 ventral root there. And in the same issue of Cell, Dan Cleveland also published back-to-back mouse NFL overexpressed with the CMV promoter. He also got large accumulation and swellings and some degeneration of axons, and this made in those days the cover picture of Cell. And we got another paper 2 years after showing that if we inject S35 methionine in the spinal cord of these mice and do a classical it's a kind of exposure of fluorograph of different sections of the sciatic nerve. You can see the bands here, NFL, NFM, NFH. They migrate about 1 millimeter per day, but the transport was blocked when there was NFH human overexpression. So this was affecting, this change of stoichiometry was affecting the transport of protein in the axon. And this is why on longitudinal section you could see that there was major atrophy of axon. The myelin thickness remained the same, but this was normal and NFH transgenic. So there's an importance of stoichiometry. If you overexpress human NFH, you get small axon, large cell bodies. If you overexpress human NFL, a little bit of accumulation of neurofilament in cell bodies, but normal, basically normal axon like the wild-type mouse here. But if you cross these two mice, we were rescuing the NFH over phenotype. Then there was less accumulation in cell body, and the filaments were able to move into the axon. So restoring in part, the stoichiometry restored the transport of filament in the axon. We did also a similar experiment with peripherin. This is peripherin overexpressor mouse. We've used either peripherin intact gene or also the Taiwan gene promoter, neuronal promoter. And the mice develop phenotype, overt phenotype, but it takes a long time. It takes 18 months to 24 months to get phenotype. But the overexpression of normal peripherin by fourfold, it can kill motor neurons. It's just a matter of time in aging. And peripherin also got large accumulation in the axon, not only in the cell body. And that's probably why it was more toxic, the overexpression of peripherin. The accumulations are not only in cell body, it blocks the transport of material. And when we overexpress peripherin in a context of NFL knockout, everything was faster. At 6 months, we could see degeneration. There was 35% loss of axons and motor neuron. But if you cross these mice to NFH overexpressor, there was a sequestering of peripherin in cell body, and there was no more depth of the motor neurons. So you see again that the stoichiometry made a big difference between the outcome and viability of nerve cell. And we've done also real-time imaging to look at the imaging of transport of different organelles in the DRG neurons from peripherin to an overexpressor, and we found that disorganization of peripherin can also affect transport of mitochondria. And in culture cell, the net transport of mitochondria was retrograde, not anterograde like in normal DRG neurons. So in vivo, I don't know in vivo how this can be explained, but in vitro at least we see that there is a difference in the transport of mitochondria. Now the role of neurofilaments in the radial growth of axon, the definite proof that neurofilaments are involved in axon caliber was, I think, the NFL knockout mouse. There was also a quail mutant of NFL, a natural mutation in the quail a few years before that was shown to have phenotype and absence of neurofilament. So in absence of NFL, the two other subunits in the mouse are unable to properly move into the axon, and you get an atrophy of axon. The mice don't die, but they're not perfect. The conductivity of the nerve is reduced very much by having absence of neurofilament. That's probably because of the caliber of axon. The axons are filled up with microtubules. You can see cross-section there. You can see longitudinal section. But the absence of NFL is not always a bad thing. When we've crossed these mice NFL knockout with mutant SOD1 mice, and more recently, Michael Sedner have shown that if you cross PMN mice and other motor neuron disease mouse with NFL knockout, the absence of NFL was protective. The mice lived longer survival, and we didn't know why at those days, and we had opposite title, but basically it was protective effect of overexpression of neurofilament. Here was absence of neurofilament and protective effect, and the reason is that in both cases it was an absence of neurofilament in the axon. And I think Mike Sidner has a proper explanation for that. He found that in absence of NFL, STAT-min binds more to STAT3 protein, and STAT-min is involved in destabilization of microtubule. So basically, the absence of NFL makes microtubule more stable, and that explains why we saw more microtubule in the axon. And this is able to be protective in context of PMN and SOD1 mice. Now, what's the role of phosphorylation in the radial growth of axon? Many labs have done several experiments on that, and it was believed that phosphorylation would extend the sidearms of neurofilament and maybe control the distance between the filament and control the axon caliber. So it came as a big surprise in a paper we published in 1998 in JCB. There's Don Cleveland also and Bob Lazzarini. In the same paper, we had 3 papers back to back with a kind of review by Hirokawa about neurofilament, because absence of NFH did not change the caliber, and that was a big shock for people working on neurofilament. What's going on? And it's later on we found that NFM knockout, on the other hand, was a smaller caliber in NFM knockout. So NFM was involved, but not NFH. And of course, if you knock out both of them, the caliber is much smaller. And there was work published by Ralph Nixon here following that if you remove the tail of NFM and make a knock-in mouse with a lacking NFM tail, you can also show that there was a change in radial growth of axon. The axon was smaller, so the tail of the NFM was involved in this phenomenon. And there was work by Michael Garcia also that showed if you replace the mouse KSP repeat by the bovine KSP repeat, I think there was a change in the length of this region. This can slightly increase the caliber of the axon. So definitely the NFM was involved in the caliber of axon. So Michael Garcia did an experiment where he replaced all the KSP repeat with a KAP repeat, replaced the serine by alanine, and make transgenic mice with that, and there was no change in caliber of axon. So the KSP repeat and NFM were not involved in the caliber. It's something else, or another phosphorylation site, but we don't know exactly what's the mechanism by which NFM controlled the caliber of axon. Yeah, I think it's still an open question, this. Now I would like to talk a bit about the long half-life of neurofilament in the axon and axonal transport, a bit about axonal transport. So I think the half-life of neurofilament is a topic of relevance for the meeting today. This is how we did that. We've published this about 10 years ago, but I did not present this data very often. So I went back to my old slide to try to understand what I did in those days. And so basically we did 2 transgenic mice, one with the prion gene promoter driving the TET activator, and the other mouse having the TET operon driving human NFL. And you remember we have a specific antibody for human NFL that doesn't see the mouse. So we generated a double transgenic with this, and at a certain point we treat the mouse, we feed the mouse with doxycycline. And doxycycline turned off expression of NFL. So the mouse is born with human NFL being expressed. And we did that in the context where there is no mouse NFL, or in the context where there is mouse NFL. And this is the results we got on the Norton blot. This is the HNFL, human NFL messenger RNA. If we treat with doxycycline for 1 week, the mRNA disappears after 1 week. And then if you stop feeding with doxycycline, the expression is coming back again within a few weeks. If you look at Western blot in context of NFL mouse knockout, it takes average, in the cerebellum, it takes average 3 weeks for the NFL to disappear. So the half-life was 3 weeks for the cerebellum. So it's a long time. We did the same analysis, but this time for the sciatic nerve. And these are segments of sciatic nerve, 6 millimeter segment. We did the Western blot of this after 0 weeks, 2 weeks, 2 months, 3 months. And you can see the band NFL here decrease all along the spinal cord in the context of mouse NFL knockout. So again, we reach a 3 weeks average in context of knockout. But in context where mouse NFL is heterozygous, the human NFL did not disappear. Even after 3 months, it was still there. And we did some analysis here on the graph. So in context of NFL, mouse NFL knockouts, the human NFL is decreased, half-life 3 weeks. But if the mouse NFL is there, it stays basically stable. And I can tell you, we've looked at 8 months after and it was still there. So the very, very slow degradation of human NFL when there's an existing filament structure. And if you stop feeding the animal, after 2 weeks, you can see human NFLs coming back 3 weeks, 4 weeks. And they're coming back all along, in synchrony, all along the axon. It's not, there's no wave of protein moving back. It's really that in absence of neurofilament, it just moves very fast probably, and you see the whole nerve becomes filled up with NFL. And if you stop doxycycline, if you stop feeding doxycycline, that's what you see here with time. We did another experiment where we had the adenovirus encoding the TET activator. We injected this virus in the tongue of animal that has the TET operon driving human NFL. So the experiment, we wanted to know how fast they would be transported. So the virus goes back to cell body in the hypoglossal neurons, and they get synthesis of human NFL. And we expect that in the hypoglossal nerve, we got the proximal and distal 6 millimeter segment. We expect to, at 1 millimeter per day, it should be, if it moves with slow transport, it should be there within 6 days. But we found that in absence of NFL, they were there after 2 days. We can detect here the human NFL. And after 5 days, almost all the human NFL was in the distal. So the speed of filaments moving was about 10 millimeter per day, not 1 millimeter per day. But in context where mouse NFL was there, it was 1 millimeter per day. So we came out with this schematic representation about transport and stability of neurofilament, where axons with a low content and axons with a high content of neurofilament. In a low content, the filaments are moving faster than in this one, which is full of filament. And the proteins are also degraded much faster. Even though it takes 3 weeks, it's degraded faster. In the context where they assemble all along the way, they're very, very stable. So that's the conclusion from these experiments. I would like also to present one slide on this thing, because it's going to be published within a week or two. I know it's in press. It's a collaboration with Tony Brown, and it took a long time. About 10 years ago, we generated transgenic mice with him. This is a Taiwan gene promoter driving NFM that has a photoactivatable GFP. So that if you illuminate with UV light, you can label the NFM, and you can follow how the NFM fluorescence disappear. And this is what he did there in the internode region, which are full of neurofilament, and did it for the node of Ranvier here, just photoactivate the NFM, this region. And he found that in the node, the fluorescence disappear very fast. In the internode, it's much slower to disappear. The filaments are moving in the adjacent region of the place you do the activation. And he concludes that neurofilament must move much faster in the node of Ranvier than in the adjacent region, which makes sense because otherwise filaments will pile up in the proximal or distal region. So that's what you will find in this paper that will appear very soon. I thought it would still be out by probably next week you'll see it. Now, neurofilament involvement in neurofilament disease, in human disease, it was known for a long time that neurofilament accumulations were detectable in ALS, and they were called Lewy bodies and axonal spheroids by neuroanatomists. And in this case, this is an anti-peripherin antibody, but you get similar picture with anti-NFL and anti-NFH. And early on, we detected that there was polymorphism in the phosphorylation domain because of the KSP repeat. I thought maybe there would be some variation in human. And indeed, we found that there were two alleles in human, the 44 repeats or 45 repeats. And this led us to screen for this region in ALS. We did this in collaboration with Denise Figurowitz, who was a postdoc at that time with Guy Rouleau. And Guy was his lab adjacent to mine, so we decided to collaborate on this project. And we found a few mutations. We found a lysine that was lost in 4 patients, and 1 patient with one of the repeat sequence that was deleted. But there was no family. We didn't find any family. They were all in sporadic cases. And there is another researcher here in London, Al Shalabi, and Tompkins also. They found other deletion in that domain, the KSP repeat deletion. There was 1 case of insertion. I think it's Tompkins published that. All others were deletion, but again, there were no families, sporadic. And Shalabi did another paper, published another paper about the 2 alleles, he called the long and the short allele, and found that the short allele was more frequent in motor neuron disease patients than in the control, 50-37. And people homozygous for the short allele frequency was 33% versus 6% for the control. So he sees the short allele as a risk factor for ALS, which corroborate what was found with all the others today. The NFM and NFL sequence, some people have screened Don Cleveland or ourselves and didn't see any variation in ALS. On the other hand, the NFL mutation can cause a Charcot-Marie-Tooth disease, and it can cause neurofilament accumulation along the axon, large accumulation. And several mutations have been discovered in NFL. It was first published by a group in Russia. And the mutations were found to cause aggregation of filaments when you express them in culture cells. So there's definitely something wrong about the assembly of NFL in the presence of mutation. So the conclusion to date from the transgenic mouse studies and genetic screening in human is that alterations in stoichiometry of neurofilament intermediate filament, neuronal intermediate filament, can be detrimental, can form aggregates, axonal transport defects, change in caliber of axon and conductivity. And you will hear today from Ralph Nixon about change in glue and 1 subunit also. And something nobody is talking about, but neurofilament can bind calcium. And I'm pretty sure that change in neurofilament level can affect calcium homeostasis, especially in motor neuron. There is a lack of calcium binding protein. And if you remove neurofilament, I'm sure it will contribute to the increased level of calcium. And another conclusion is that neurofilament gene mutations can cause disease, but they are rare in ALS, not a major cause of ALS. In ALS, it's a disease that involves degeneration of upper and lower motor neuron. Life risk is 1 in 1,000 to die of ALS. The majority are sporadic case, 90%, 10% familial. There's no effective drug really for ALS at this time. In the past decade, several genes have been identified. The first one was in 1993, superoxide dismutase. Then came human NFH, and several other genes coding for cytoskeletal protein, RNA binding protein, and protein degradation. So we end up with a disease with different genes having different function causing a similar disease. But there are something in common. In most patients, there is an accumulation of a protein, TDP43. The marker of disease or neurofilament, I would say, TDP43. And TDP43 is mainly present in the nucleus normally. It's involved in the splicing of genes, many RNA, splicing of RNA. And the knockdown of TDP43 in the nucleus can cause aberrant splicing of several protein that was published by the group of Phil Wong in Science 2015. But in the disease, there is a loss in the cell of the TDP in the nucleus and a gain of function also aggregated TDP in the cytoplasm. And this also contributes to the disease, not only the loss in nucleus, but also the gain in the cytoplasm. So the protein schematic representation as nuclear localization signal, RNA binding domain, and the glycine-rich region where most of the mutation have been found. I think there's 35, 40 mutation in TDP43. And as soon as this was published by Kuhle, we generated transgenic mice expressing 2 of these mutations. And the mice developed with time. So the way we did it was using a genomic fragment, 20 kilobase genomic fragment. So the TDP gene is expressed with its own promoter, introns are still present, and so on, C-terminal. They just carry different mutation. And during aging, at 2 years of age, 1 year of age, they start developing accumulation of human TDP in the cell body. So they are a model of TDP43 accumulation in cell body. In motor neuron and cortical neurons, they develop that. But they don't die of this. Somehow they resist to this, not like in human. But still, it's a model, we can do something with this. And they also develop peripherin accumulation. This is in spinal cord accumulation of peripherin in cell body. On Western blot, you can see a huge accumulation of peripherin. Also the splicing variant. And many years ago, Janice Robertson, who is now at University of Toronto, showed that in ALS, human ALS, there's an increase of a variant of splicing variant of peripherin, PER61, which is, this is also induced in these mice. So there's an accumulation of this PER61 that they stain also antibodies with anti-PER61 antibody you can stain, similar than in human. Neurofilament NFL is down in these mice. And this is in the spinal cord analysis on Western blot, transgenic expressing mutant TDP. This is a normal mouse. And so this fit pretty well with the work done a long time before 1994 and 2000 that by in situ hybridization, Bergeron and also Michael Strong showed that the level of messenger RNA for NFL is downregulated in ALS patients in post-mortem analysis. So the messenger RNA for NFL is downregulated. So our results fit with that. But Mike Strong also showed that TDP43 can bind NFL messenger RNA. And these are the binding sites. But the TDP is stabilizing NFL messenger RNA, not destabilizing. So, and he proposed that maybe NFL messenger RNA can be sequestered in the stress granule, because there's also stress granule formation with TDP43 in the cytoplasm. How can we explain the change in neurofilament level? I'll come back to this. So. What we found is we were searching for a way to alleviate the TDP43 pathology. And I'll show you we can do it with a compound called interferon A analog that we call IMS-088. I will explain about that. So this project started with the finding that TDP43 can interact with NF-kappa B subunit P65. So in the innate immune response, NF-kappa B65 is the subunit that goes to nucleus and activate genes that are involved in innate immune response. And TDP43 in ALS samples, spinal cord sample, but not in control sample, can pull down. If you pull down TDP, NF-kappa B was pulled down with it. So demonstrating there was a direct interaction. And in transgenic mice that we've produced, we saw this interaction as well. And this led us to think maybe we can try a compound that can inhibit NF-kappa B signaling and see what will happen to TDP and so on. And there's a compound called ashwagandha, which is an extract of a plant called Rhytania somnifera. It's a plant that has been used for 4,000 years in India to treat all kinds of disease, anti-stress, anti-inflammatory, rheumatic pain and arthritis. And you can buy it on the internet. It's very cheap and no much side effects. And there's a compound in this plant called interferon A which is a steroid lactone. And we decided with a small company, a chemist, to modify this compound. We tested about 30 different derivatives, and we came up with this derivative, which was better than the original in terms of less toxic, but it's still doing the job as NF-kappa B inhibition. And this is the compound that we tested first in culture cell, 293 cell, where we can induce TDP43 pathology. You just treat the cell with an oxidant called itacrinic acid, and itacrinic acid makes the TDP going in the cytoplasm forming aggregates. So we treated the cell with the IMS 088, and the number of aggregates was reduced. So in culture cell, we were able to reduce the amount of aggregates TDP. We wanted the system in vivo, so we had mice expressing human TDP43 mutant. And we treated with this compound orally at 30 milligram per kilo for 6 weeks. And this was good. The performance of the mice, cognitive performance was good. On the novel object recognition, the TDP mice do not recognize the new object, do not distinguish new object from old object, spend as much time. But following treatment, they recognize the novel object, so they have better cognitive performance. And we look at the aggregation. You can see a punctate structure of TDP43 in the cytoplasm here. And following treatment with IMS, TDP become nuclear, and that is in cortical neurons. And we've measured that. There's less cytoplasmic TDP43 following treatment, less insoluble TDP43 following treatment in the 2 transgenic mice, different mutation. In the spinal cord, we saw the same phenomenon. This is in the untreated cytoplasmic TDP. This is treated, it's in nuclear TDP, and there was a major change in the distribution, lower cytoplasmic versus nucleus. So now, this is where it comes with neurofilament. We decided to look at the translating ribosome. We made transgenic mice with the neurofilament promoter driving ribosomal protein, which is tagged with HA, hemagglutinin, and the red fluorescent protein. So the trick is that once we have this mouse, we cross it with TDP mice, and when they adult mouse, we can immunoprecipitate the ribosome. And we can analyze what's the proteomic, what's the peptide that are being made, what's the peptide being made. This is to show it was neuronal expression, only neurons express this thing. So we can analyze the peptide which are being synthesized in the ribosome. And we came out with a very surprising results. If you look at the A panel here, this is the TDP43 mice versus a normal mouse. We found that the analysis of proteomics, we had about 1,200 protein. A majority of protein, there's no change in the translation. But neurofilament protein are highly downregulated. They're regulated by 4 to 5-fold NFL, NFM, alpha internexin. They also RNA binding protein are downregulated. Also proteins involved in neurite outgrowth. Protein involved in apoptosis are upregulated on the other end. Now in B, it's the panel that compared the TDP43 treated with IMS versus the untreated with IMS. And the IMS treatment completely restore expression, synthesis of neurofilament, NFL, and alpha internexin, and downregulate this time the deleterious gene. There was major change, and this was confirmed by Western blotting of cortical of brain neurons. This is total extract of brain. Western blot you can see with vehicle and with IMS treated. And so in summary, we found that this compound was able to ameliorate behavior, to restore the mislocalization of TDP, and to restore the abnormal TDP, the abnormal level of TDP in the cytoplasm, and also restore synthesis of protein, synthesis of neurofilament protein that was reduced in this mouse by 4-fold. And we now would like to go into clinic with this compound, but there's still things to do, a different dose with pharmacokinetics, toxicology, and we need to produce large amount, 1 kilogram of this compound before starting clinical trials. So it will take another year, I think, before we're at that stage. And for the discussion, based on what I presented, I think for the biology and function of neurofilament, the highly phosphorylated protein play a role in axon caliber. Change in stoichiometry can cause deleterious effect. They have very long half-life. Axonal content can influence axonal transport and degradation of neurofilament. And in fact, little is known about neurofilament degradation in vivo, and it's something Pascal Beaumont will address this afternoon. We'll keep it for later. For ALS, neurofilament accumulation, they are all mark of ALS, and maybe other disease as well, but also in ALS. And the NFL messenger levels are reduced based on in situ hybridization, but TDP, which accumulate in the disease, can bind an NFL messenger RNA to stabilize it. And it can also block the neurofilament messenger RNA translation. And I would like to acknowledge the people who contributed to the last part of my presentation with IMS and neurofilament, Sonny Kumar, Daniel Faneuf, and on the TDP, Carol Duta. And I collaborate with my wife, Yasna Kriz, who has generated the ribotag transgenic mice. Okay, thank you for your attention.
Yeah, so Jean-Pierre, if you wouldn't mind taking some questions, I think this is the most interesting time. Let me just start with one just to kick things off. Many of us who've been interested in plasticity are trying to understand the plasticity of the axon, activity-dependent changes. So can you describe what is known about the role of intermediate filaments in controlling this plasticity-dependent change in axon diameter, and what the mechanisms regulating that might be as far as we understand?
Yeah. Well, during development, the NFH gene is not expressed. It's expressed postnatally. And during development, very early on, neurofilament is not there. It's nestin and vimentin, which are being replaced then by NFL and NFM. And postnatally, the NFH occurs once the synapse is formed. So it could be that NFL and NFM are more involved. You need that to have a faster growth, and once it's growth, the NFH would stabilize the structure. This is how I view it. During a nerve regeneration following axotomy, neurofilament, all three neurofilaments are down regulated. Peripherin is up. And this maybe it's a change in the stoichiometry of filament that's allow the axons to regrow as fast as possible. And then the neurofilaments are on again. In terms of TDP43, following a crush, TDP43 is moving from the nucleus to the cytoplasm. And once the nerve has regrowth, it's coming back within 4 weeks in the most to the nucleus. So it could be that it's involved, and now that we know it's blocking the neurofilament NFL, maybe that's what is responsible for the block of synthesis. Never thought about this. Just coming to my mind that maybe that's the mechanism. I should put a student on that when I come back.
So that model would place neurofilaments as part of the long-term stabilization mechanism. Actually militating against rapid changes in axon diameter, and therefore, for example, induction with changes in metabolism.
Yeah, it's a way probably to make. It has a role is the maintenance of axon also. The NFL knockout mouse, they're very fragile. The nerve. If you can crush this, they're not very resistant. And in large animal species, I'm pretty sure that would be disastrous. And human, there are some cases of Charcot-Marie tooth in human with frame shift mutation in NFL, and they're not doing very well. I think it's pretty severe Charcot-Marie Tooth case. You can imagine a whale also with a 10-meter long axon. They would take a whole life for filaments to reach the nerve ending. So it must be very, very stable, and it's there to remain as it is. I think that's how I see it. Even though they have movement, if you do S35 methionine pulses, there's probably some change, but probably minor. The system is made to have it stable. Yeah. But that may vary in some cell versus other also. And you look at many cells that degenerate in nerve cells that degenerate in human disease, Parkinson, Alzheimer, ALS. They're relatively large cells that contain lots of neurofilaments. And maybe there's kind of correlation there. Risk factor. Yes?
So following on from that, do you see any relationship then between individual motor pathways, for example, and the expression or loss of that geometry in neurofilaments?
You mean in terms of ALS disease?
Well, yeah, exactly. Sorry, so the TDP-43 is problematic because it doesn't die. But in terms, do we see regionality?
They don't die suddenly, though. They can go to the cytoplasty DP, and it will be a slow process, that degeneration.
But in terms of sort of, is there any indication that we see a relationship between the changes in the stoichiometry and neuroanatomical pathways that we know are affected? So in the lower motor neurons, upper motor neurons, brainstem nuclei, etc.
Yeah, I don't know about this. I don't really. I don't know if anybody has done the study on this stoichiometry versus how in vivo the change in stoichiometry. We did it with transgenic way, but in a normal life, all these changes occur. We're not certain. There may be intoxication. There are many drugs that can change neurofilament accumulation, for example. There's Exane. What's the other thing that? Yes, exactly. So many other things that can cause problems. But we don't know if it's for the translation. It's the first time I see there's something occurring in the translation. So maybe this is of relevance to your question. Not very clear. Yes, Pascal.
I have several questions, but some I can save after Ralph's talk, so that it's timely. For the stoichiometry, so you work with different models of expressing knocking out all the genes, and it's really complicated story. Do we have, at the end of the day, an idea of the exact stoichiometry we should keep in human and in mouse? And do also we have an idea if the fold increase that you show in the different models or even other groups correlates with this?
Yes, I think the stoichiometry in the mice is about 532 for the NFL, NFM, and NFH. And almost anything you do, you will see some change in the small accumulation of filament in what I see. Heterozygous mouse, they don't have much. So if you change slightly, it is not highly changed. But we did once a transgenic mouse with 3 heterozygous, NFL, NFM, and NFH, after level of neurofilament, but without changing the stoichiometry. And the caliber was smaller. It was just less filament in the axon, but the stoichiometry was perfect, so there was no problem to this mouse. But as soon as you overexpress one versus the other, there's a problem. And the mouse also NFL is different than human NFL. The mouse NFL require an NFM and NFH, or maybe alpha-internex to form filaments. Otherwise, it doesn't form filaments by itself. In culture cell, it doesn't form filaments by itself. But a human NFL does. So it could be that there's a small change in the biology, an assembly of NFL in human versus mouse. Good at that.
Yeah. Speaking about stoichiometry, but also referring back to your comments about the severity of Charcot-Marie tooth disease, it kind of contrasts with the fact that when you knock out NFL, the motor phenotype is actually very mild and in fact imperceptible unless you're measuring very sensitively. And even if you knock out all 3 neurofilaments so that you lose all 4 neurofilaments in the CNS, you get a phenotype that is measurable at the level of behavior and synaptic function, but you would not be able to discern a motor deficit without doing very sensitive changes. So you can eliminate all of the neurofilament subunits, and this would suggest to me, and maybe this is my question, as to whether the CMT and some of the mutations are gain of function and related to this notion of imbalance, so that you are throwing off the imbalance. It could be some other gain of function explanation as well, but rather than just a loss of neurofilament subunit, it's a gain of function due to some other aspect of neurofilament.
Yes, you're right that the loss of neurofilament is less deleterious than the mutation neurofilament. But this was observed also for the keratin genes, that the loss of keratin genes has less deleterious effect than the mutation. There is a gain of function of the mutation. For neurofilament, it could be that it can be a factor that provoke accumulations of filaments in the cell body or axons, and these aggregates can have a level of toxicity that more than having no neurofilament in the axon. That's how I see it. In the field of keratin, I think this is also what's going on. Accumulation of aggregates in cells can sequester other proteins involved in these complex network of intermediate filament, and it makes things worse having the mutation, mutant protein, and the absence.
One other quick question, as long as I have this microphone. The drug that you're using in your company, what effect does it have on TDP-43 itself? Does it alter? You say it restores the neurofilaments.
Yeah.
Does it have any effect on TDP?
The evidence we have is that by inhibiting NF-kappa B, it pushed the I-kappa B kinase towards autophagy, increasing autophagy by phosphorylation. So the I-kappa B kinase is using other substrates which are involved in autophagy, and there's more degradation of TDP-43. So in the cytoplasm. And since there's more degradation in the nucleus, there's a slight decrease in nucleus, but basically you restore more normal distribution of TDP in the cell because of that. So it's a question to do with the NF-kappa B pathway inhibition that can also lead to autophagy of the accumulated protein.
A question, or a comment and then a question. A comment related to Ralph's first question on the role of, why isn't the mouse worse off? If we're talking about increasing susceptibility to injury, whether it be physical or other kinds, a mouse in a cage may not be the same as a human athlete or a whale or whatever. So this whole idea of strengthening the stability of the neural morphology and its interaction with other cells to make tissue may be an important factor. So when you put a stress on, that's when you may see more of a mess. The question relates to the TDP-43 story and whether the effect of the with alpha or the IMSTAR 088 is simply a relationship to, you know, it keeps TDP-43 in the nucleus and everything can proceed as normal, or whether there are other things, and whether there's any also an effect of mRNA retention in the nucleus because of cytoplasmic transport, as well as the translational. In the mutant FUS model, we see both. Not with neurofilament, we haven't looked. So that's the question part.
Yeah, we haven't looked at NFL, messenger RNA in the nucleus and splicing and things like that. We just looked with the ribosomal. We were not interested in neurofilament when we did this experiment. We wanted to know the change in what was going on in protein synthesis, and it's neurofilament that showed up. The major one are neurofilament. And this fits with is going back to ALS with neurofilament accumulation from 30 years ago, 40 years ago. Yeah, exactly.
So Jean-Pierre, related to the disease, I always wonder, we always assume that NFH polymorphism, it's considered as a risk factor for ALS. And I wonder, does anyone really try to look at the functional effect of this variant? Because
a long time ago,
really
we did transgenic mice. The one we've published in Cell was the low repeat. We didn't know at that time there was a difference. We also did with the long repeat, 45 repeats. And the mice, they did not develop more than the 43 repeats. So it was the same, so we probably still have the embryo frozen, but I stopped analyzing them. So there was no difference from what we can see when you put, even the mutation, we put human mutation in the mouse, and they were behaving like the one we've published with the normal allele. So probably the system is not good enough to pick up a difference in the mouse.
So it's still an open question then.
Yeah. You know, we're dealing with a late onset disease with subtle change that occurs all along the life. That environmental factors may be part of it. And it's something that can be hard to reproduce in mice. And there's no perfect model of the mouse for ALS at this time, as far as I know. Even the SOD1 is probably an exaggeration because there's 25 copies of the SOD1 express, and which is already a high expression gene. So, yes. Okay.
You will get the mic. So.
He has the microphone. So he has
when you look at expression data, from your perspective, is there any hope of neurofilament light biomarker that would differentiate between central and peripheral nerve injury impairment? I mean, peripheral has this seductive name, but
yes, I don't know. I would take alpha-internexin then to look at central versus peripheral. Would be peripheral.
You mean internexin versus
for the CNS?
Yeah.
CNS would be internexin.
Very minor expression. It is surprisingly expressed in some of the bigger nerves, especially optic nerve, but it is and very low, almost vanishing amounts in most of the CNS. Yeah.
Otherwise, I don't think we can distinguish. There's no major difference as far as I know. No. Yes.
You mentioned with the drug that you're developing that it restores the neurofilament RNA, if I understand correctly. Do you therefore expect that a positive treatment effect would increase the neurofilament concentrations? Because you normally assume that the levels decrease as a treatment response.
I don't think it will increase because it's the TDP in the cytoplasm which is blocking the synthesis by binding to the 3' of the messenger RNA. And if you remove TDP, it will come back to normal unless there's more RNA. It could be there's a bit more RNA at the time we treat with the drug because there's a sequestering also in the cytoplasm, probably of RNA. But I don't think we will increase the level of neurofilament with the drug. In normal mice, we didn't see that.
Thank you. you. I was interested in your red fluorescent protein model showing the transport accelerating across the nodes of Ranvier. Have you looked at how further downstream it may be transported out of the synapse and how that is affected in these kinds of nerves? Because it may start to give us some information on the relationship between a peripheral fluid biomarker, so what's coming out of the cell after these changes with disease.
You mean the transport of what protein?
So the neurofilaments.
Of neurofilament after a treatment?
Out of the cell.
Yeah, that would be very interesting to look at. No, we got these results relatively recently. It's not published yet. We're writing the paper, but that would be a very interesting aspect to look at what's going on in the transport and so on. Yeah, absolutely. And also the exact mechanism, how it blocks the translation, I think would be something to study.
Just one other quick question, Jean-Pierre, before we move on. Can you comment on... so I'm just curious about... I've been thinking about the axonomy model. And when you snip an axon and there's partial dying back peripherally, but then regrowth, what actually is happening at the interface between, what is an existing stable intermediate filament structure, and then one has something that's degraded distally? And as the growth cone starts to move forward, is that intermediate filament that's filling in, or is there a recapitulation of developmental processes with Biomarker and other?
I think what's going on is that the distal portions get degraded pretty fast by the macrophage, and the macrophage also invade the dying axons, and they produce a growth factor and for the axons to regrow. And if you deplete the macrophage, and we did this experiment about 10 years ago, we had, it was thymidine kinase we treated the cell with the suicide gene, basically, so that the macrophage were not present anymore in the distal tip of injecting a drug. And the nerve do not regenerate well if you do this. So you need, the macrophages are there, they generate nerve ending, and then they can grow. And they contribute also to provide a growth factor.
And at that stage, so I can see, that sounds like the macrophage is needed to, to clean up the structures, the messed up structures that are left by calcium influx and whatever else happens distally.
Yeah.
But then in terms of what the physical substance that is replacing the scaffolding for the growth cone, is that more, are those early proteins like Biomarker, or is that actually intermediate filament that's being passed down the axon in order to fill in?
I think the tip is probably more devoid of neurofilament. It's the microtubules and the dynamics of microtubule and actin that are pushing the axon, and the neurofilaments, I think, are stabilizing after they regrowth. This is how
so they're filling in at some later stage, which allows
or they can be made up in the nerve ending also. They can be synthesized there. RNA it can be transported and maybe synthesized on site. I've seen some paper some years ago about that neurofilament proteins can be synthesized at the tip of the nerve ending.
But if I may, there are data showing that. So there are data showing that I don't know about the process, who is coming first, as you mentioned, the developmental or the adult stage. But what is known in at least one paper is that if you deplete mouse from neurofilaments, you impair regeneration. So which implies that it's not only a scaffolding, but it's really required for I don't know what mechanism to occur for regeneration.
But in vivo, the ENFL knockout mouse, when we did a nerve crush, they do regrow. They regrow very well, no problem. But the myelination is retarded because the axons are smaller in caliber, and you need at least a 1 millimeter to start myelination.
As I was mentioning, I don't know if you remember the NFH-LAC-Z overexpressing mice,
Okay.
where
Or
the
legs,
neurofilaments
yeah.
are stuck
Yeah.
in the soma, so then the axons are devoid of neurofilaments.
Yeah.
But it's a different impairment, but it's still
But
impairment
yeah, it's a different
myelination.
mouse, yeah. So they do regenerate, but I think the speed must be about the same. From what we did calculation, it was very tedious experiment to do this. But they were regenerating well, but myelination was retarded because the caliber was not restored.
So you said in acute injury that L and M are downregulated. So do you believe the higher levels we measure in the body fluids are really degradation products, or are they upregulation, compensatory? Or because if you say they're downregulated, it's an argument that they may really come from degrading nerves, right? But what's your opinion on that?
I don't know. I think Jerry Shaw did some experiment on that some years ago about doing spinal cord injury and measuring. But these were spinal cord injury. It can be interpreted different manner, but this is how he showed the first time, I think, that neurofilament can be used as marker of degeneration in the CSF. And I don't know about this.
I think many of the studies, we haven't done studies of regeneration, but the studies suggest that there's an initial downregulation of expression, but then when the nerve starts to regenerate, there's an upregulation. So I guess it depends on what phase of the process you're talking about. The degeneration is occurring very quickly, the regeneration more slowly, so you might see a reduction early, but in a mixture of nerves that are regenerating or degenerating, you're going to have sort of a combination of higher expression and still suppression of expression.
Well, Jean-Pierre, you've obviously catalyzed an enormous amount of discussion. This has been a fabulous way of starting. Thank you.
Thank you.
Okay, well, this was a great discussion, and this is exactly what we want the day to proceed as. So please, everyone continue to contribute. So our second speaker, has already shown us what energy he injects into the room, and he needs no introduction. Henrik Zetterberg from Gothenburg and UCL.
Thank you very much. Yeah, this was a wonderful start of the day. Now, let's see, I should start my presentation. There she is. Okay, yeah, good. I couldn't find it either. Thank you very much. Yep, so now we will move into the fluids and the biomarkers, and we will see how we can correlate what you just said, Jean-Pierre, with what I will show now. We will also hear more about this from Jens Kuhle later, and we divided it like this, that I talk more about neurodegeneration and injury, traumatic injury, and Jens will tell you a lot about multiple sclerosis and also some other diseases. And we will try also to see what data we can present on dynamics in response to treatments, because that is an important aspect today in our research field. So this is just an updated slide on what types of targeted biomarker assays we have now that work on CSF and to some extent also on blood lately. So we can measure targeted a lot of proteins from synapses now, SNAP25, SUT1A, and we also just have a method now for SV2A. You all know that there is a PET ligand for SV2A, but now we can also measure it in a fluid. We always struggle with interpreting fluid biomarker results, just really because of what we just discussed. Is it increased expression or release from degenerating structures. Why are levels changing in a fluid? And here we need to do more translational work together, I think, in the models. Tau, we thought for a long time, was a neuroaxonal injury marker, but there is a lot of data indicating that that probably was a misinterpretation, at least in Alzheimer's, where it instead looks like exposure of neurons to beta amyloid or something else increases the expression, release, and phosphorylation of the protein from cells that are still alive. So it's not a direct marker of neurodegeneration, but a neuron secreting a lot of tau and phosphorylated tau will probably eventually degenerate. It's a surprisingly AD-specific biomarker. Neurofilament light stands out as our general neuronal injury or neurodegeneration marker, and I will show you quite a bit of data regarding this. The neurofilament light data I will present were all generated with sandwich immunoassays with antibodies targeted against the rod domain of the protein. They are specific there is no cross-reactivity with M or H here, but it has really been challenging to develop assays that extend N-terminally or C-terminally on the protein. And many of the indirect evidence suggests that what we are measuring is a fragment and potentially also a little bit of an oligomer of neurofilament light. Phospho-tau we thought was a tangle marker. It's probably an indirect marker of tangle pathology and more a direct marker of phosphorylation of tau. Then there are a lot of proteins in neuronal soma. We have neurogranin as a dendritic marker. But I know Ralph will present neurofilament light in synapses. I mean, so we really have to be careful when we interpret what these markers actually do represent. Astrocytes and microglia shed a lot of things into the CSF, and we have a lot of APP and A-beta markers and one blood-brain barrier marker in the fluids. Of course, we need more blood-brain barrier markers. The CSF serum albumin ratio is really, it's an old marker, but it works reasonably well in infection and inflammation. And then we have no really good markers in the fluids for TDP-43 pathology. It's such an abundantly expressed protein, but we can measure TDP-43, but it probably reflects general TDP-43 synthesis. Alpha-synuclein has a similar problem because alpha-synuclein is so abundantly expressed, and we have no Levy body pathology marker, except for the recent seeding assays that have been published, both from Edinburgh and from Claudio Soto's group, where the results look actually promising. But this talk will focus on neurofilament light now. And we started to work with neurofilament light from an injuries perspective. And one of the first studies I was involved in when starting in the clinical neurochemistry lab in Gothenburg was a study where we approached people who voluntarily exposed themselves to brain injury to look at dynamics of the markers. It was a challenge to get this study going. I called the Swedish Boxing Association and they told me to look for car accidents. But then eventually in Gothenburg there is a local boxing club which is pretty good. And Olof Johansson is the head of that club, and he is a big guy, a previous boxer, and he is interested in all aspects of boxing. He has written the history of boxing in Encyclopedia Britannica, I think. So he said that let's do this study together. I asked him, do you think I could do CSF sampling on your guys? And then he said, if I tell my guys to do this, they do it. And I did not tell that to the ethics committee. I wrote that. Of course, they all were volunteers and signed this. So what we managed to do was that we managed to include 14 amateur boxers, and they were good. They were all trying to qualify for the Olympics. So they were high-level boxers, and then we had 10 of their friends. The only difference between the boxers and the friends was the marijuana smoking habits, which was higher in the friends. These are really straight-edge people, leading very healthy lives, most of them, and training extremely hard, of course. So what we did was that we made use of the previous literature on stroke And when we have looked in after stroke, the peak increase of neurofilament light and tau after a stroke is not acutely. It's about 7 days, 10 days. So what we did was that we took in these boxers 7 to 10 days after their qualifying game they took part in before. And then they came back for another CSF sampling 3 months after no boxing. After they had, during the summer, they didn't box, but they trained. They didn't spar. And these are the results. So here we have controls. Here we have the boxers after a bout and then after the summer rest. And you see that the boxers, they really shoot off in neurofilament light. And these increases are not, no one was knocked out. This guy was a little bit groggy, but he could compete the whole game. But here you see, these levels are from a clinical neurochemistry perspective when we look at clinical samples from MS patients. And I mean, they are here somewhere. So some of these boxers increase a lot in a manner that we think is clinically relevant in other diseases. GFAP looked like this, increased after bout, and then it went down. Tau had a similar pattern. I mean, Alzheimer's patients are up here, but yeah, quite clear changes. But neurofilament light really stood out, of course. We think this is a little bit interesting, not just, of course, boxing, everybody. Also the boxers think boxing is not good for the brain, most of them. But to me, this indicates, okay, this is a dynamic marker. This is a marker that moves in response. And I think that's important from studies we are doing when we try to find better treatments for these diseases. We didn't find any evidence of Alzheimer's pathology in these people. So, I mean, phospho-tau was normal, nothing happened. There was no, one could imagine that these quite high-level boxers would have deposition of beta amyloid and reduced amyloid beta levels in CSF, but that was not the case. The Swedish Boxing Association, they got so angry with us, so they said that we had done an unethical study, and that we also said that their members were brain damaged, and we never said that. We said it was signs of CNS injury. So we had to do it again. We got help from the medical team at the Swedish Boxing Association and did another study and got the same results virtually. The high levels of neurofilament light after bout and no evidence of A beta or tau pathology, at least not from a fluid biomarker perspective. Here is our first and only knocked out boxer. So he came after we had published these studies. He competed also. He competed up in northern Sweden in heavyweight, and then he had a knockout. And then he approached us and asked if we could come and do CSF samplings, because first he thought it was a little bit of a scary knockout, and then he didn't want to compete until his neurofilament light levels were normal. So he came 10 days after the knockout, and we got the ethics committee to approve, and then we performed CSF sampling, and he had very high abnormal levels. Then 2 months later he came back, still very abnormal. 4 months later he came back again, still a bit abnormal. Here he got tired of us. He said, "Now I don't care about your biomarkers anymore, so now I start again." But then he broke his thumb, so in the first round, so it was not really. And then he came back, and then it took 8 months for the biomarker to normalize. So it is really, I mean, it's not a super fast biomarker, and I think it relates a lot to what you have presented earlier, Jean-Pierre. But at least one would, if we have a drug that slows down a neuroinjurious process, we would expect a lowering of neurofilament light, of course. And here, now Jens will show a lot of more data on this. This is from our Gothenburg team before and after treatment with natalizumab, where you can actually normalize after 6 to 12 months CSF levels of neurofilament light. This is another study which is not published, and it's a little bit opportunistic, and this is a case series, but I still wanted to show it to you, because so these are also clinical neurochemistry data, lab data from our lab in Gothenburg. And then we have an HIV researcher who is focused on CNS effects of HIV infection, and he sent me these 3 cases. So he's doing a longitudinal study of CNS changes in HIV infection. And this is a primary HIV infection. And what happened was that this person came in, 44 years of age, with very high plasma HIV RNA levels, high CSF HIV RNA levels, CD4 cell count still normal, and then one does nothing. I'm not an expert on HIV treatment, but I think it's active expectance. But then CD4 cell counts went down, and then antiretroviral therapy was initiated. And then immediately we see a very nice regain in CD4 cell counts, and the RNA levels drop to very low, undetectable levels, basically. So that's how this treatment worked. Looks all good. The CSF levels of neurofilament light, they looked a little bit like in the upper normal range, climbing up here, and then ART was initiated and the levels go down. But I mean, this is of course still within a relatively normal range. But I think it's interesting still. This is a person who came in with low CD4 cell count and started immediately on treatment. CSF levels of neurofilament light were up and went down in response to treatment and maintained stably here. You also see that in this study we have made sure to make this type of graph where you see the age-related increase in neurofilament light, because sadly there is a very clear age-related increase in neurofilament light, which probably is, I don't know what it reflects really, but age-related brain changes that make you wiser. Perhaps that's the case, I mean, if one is positive. Here, we have an HIV dementia. Of course, nowadays, people with HIV-associated dementia, there are often a lot of other things that they are experiencing in their lives, with drug abuse and all sorts of things. But here you see the person coming in with very high RNA levels, very low CD4 cell count, start on therapy, the CD4 cell counts normalize. Here you see CSF neurofilament light, it's a logarithmic scale, very high levels, but then the treatment brings down, and here you see age on the x-axis. So you see the dynamics here. The treatment really brings down neurofilament light. And here, the person couldn't maintain the therapy for one reason or another. I do not know exactly what happened. So it was a period with off therapy here. Immediately, there are very rapid changes in HIV RNA levels and CD4 cell counts. And neurofilament light actually went up there, and then the person was put back on treatment and could do that treatment, and then the levels went down again. And the cognition also restored here in this phase. So it's really, I mean, it's a reversible dementia then. And the biomarkers change. So again, this to me, it indicates that neurofilament light is a dynamic marker that can be used to follow treatments. And here, these are also new data from also the clinical neurochemistry lab routine in Gothenburg. So these are kids who start treatment on Spinraza, nusinersen, in spinal muscular atrophy. I mean, that's a remarkable treatment that has come with the antisense mediated stabilization of SMN. So, and you give this treatment intrathecally. The time points here are weeks, and you start, when the kids start off very high in neurofilament light, and then the treatment will bring down the neurofilament light concentrations. And I didn't know that he was sending the CSF to us. So when I got to know that, because of course you can always take out a little bit CSF when you do the injection. So then we looked at these data, and I thought they were wonderful. But then he said that, well, of course he sees clinically on the kids that they are much, much better. But look at the biomarker, I said. At least, yeah. So he thought for some strange reason, he thought the clinical thing was more important than the biomarker change that I was so happy about. Yep, I think he means that he doesn't need this really. But again, it shows the dynamics of the biomarker and what we should expect in diseases where we know that neurofilament light is high. We have tried also to make blood tests for many years. We tried to do plasma A beta tests and then tau, but then with often the biomarkers, the CNS-derived biomarkers, are present at quite low concentrations in blood, and ELISA will not work. Most often that's the case. But a few years, it's almost 10 years ago, I think now, the single molecular array technique became developed, and it's a bead-based technology. So it's still a sandwich immunoassay format, but you have the capture antibody conjugated to magnetic beads, and you incubate then beads, antibody sample, detector antibody, and then you pull down the microbeads into microwells with a magnet. And the microwells, they hold a volume of 50 femtoliter, and one bead will fit into one well. So then if you dilute the sample so that the most, after you have added the substrate for the enzyme with which the detector antibody is labeled, then you can monitor light emission from individual wells. And if you have diluted the sample so that most wells do not emit light, the individual wells that do actually represent most often one analyte. So you can count molecules, which gives you a very high sensitivity. And but you can also capture the signal analogously. You could detect if the one well emits twice as much or 3 times as much. If most of the wells emit light, then you get also a quite good upper limit of detection. So the dynamic range of this type of assay is very large. And that has worked for many of the biomarkers we have tested. And it works well for neurofilament light. So here we took the ELISA, the human diagnostics ELISA. We simply took the same detector and capture antibodies and the standard protein and put it on the Simoa platform. And then we got a quite nice increase in the analytical sensitivity. And then we could start to measure on plasma. And all of us have neurofilament light detectable in our plasma for one reason or another, as we have in CSF, of course. All proteins that we thought were intracellular, when we have better assays, we see that they are also a little bit extracellularly, which of course can be a bit of a problem when interpreting a biomarker signal. But that we have been through in medicine and clinical chemistry many times. For example, troponin T. I mean, when I started having it detectable, it was bad, but now we know that we have Gaussian distribution of troponin T in normal situations. And then if you do a marathon, you will end up in the dangerous, but then return to normal. Here we saw a correlation between blood, this is plasma neurofilament light, but serum works as well, so there's no systematic difference between plasma and serum. Plasma neurofilament light and CSF neurofilament light, and this has been replicated in many studies. I think Jens will show more data on this. The correlation coefficient is in different studies between 0.6 and 0.97. A little bit depending on what range of values you have in your study population. This was the first study we did on traumatic brain injury. This is severe traumatic brain injury. And already in the emergency care department, the levels are increased, but then they increase further quite dramatically. And then we didn't design the study to look for later time points, which we regret deeply now. We would have liked to have a 3-week and 4-week and 5-week time point to really look at when the maximum is. But very high levels at day 10 to 12, and then probably they should start to go down again. These are the people who survived for a 1-year follow-up. And they are down to almost normal levels, with some people being really abnormally high still. Here is an opportunistic study, one could say, a little bit like the Boxer study. But the study was designed to test an intrathecal treatment with rituximab against progressive multiple sclerosis. But the biomarker data were actually more useful to look at dynamics from our perspective. So here we have CSF neurofilament light data before and after insertion of the ventricular catheter. So that is sort of a little bit of a minor brain trauma in my view here in this study then. And we see increase in CSF neurofilament light. This is 100, 200, 300 days from baseline. So there is an increase and then it goes down. We saw no treatment effects, but I thought this was interesting still. So increase and then down, and it's replicated completely in blood. And that brings us to another question that was discussed before. We have no clue on how neurofilament light gets out into the blood. And we do not know why neurofilament light correlates so well with CSF, whereas, for example, tau does not. But for tau, we think there is an active degradation of the protein in plasma, which is quite rapid. So tau in CSF has a half-life of around 2 to 3 weeks, and tau in plasma has a 10-hour half-life. So I think tau really gets chopped up in plasma. We will see if we can find fragments that are could be. But neurofilament light does not seem to do that. At least not using the assays we are using to measure the protein. And again, it's a mid-domain, rod domain epitopes that are very close to each other. These are the boxers, the same boxers that I showed you CSF data from. When we had the assay, we did the plasma samples also, and it looks very similar. This is a study on concussion in ice hockey in the Swedish Hockey League. It was much easier to work with the ice hockey players than the boxers, because the ice hockey players are actually concerned about this. So if you are a professional ice hockey player, you can expect to have 2 concussions per year. And there are not many occupations where you sort of know that. And in Sweden, they are not paid as much as in NHL. So the Swedish ice hockey players, they do not think this is worth it, really. I mean, so they want to be able to manage this better. And we have also players who have come out now with chronic symptoms after repetitive concussions, similarly to what has happened in the US and Canada. So basically here what we did was that we asked the teams if they would be willing to take part in this study, which was a little bit of a struggle to do, but we collected 1-hour samples in the sports arenas around Sweden, and 12-hour, 36-hour, and 144 hours. And when they returned to play, most players could return after 6 to 10 days, which is probably a bit early from a biomarker standpoint. But here, these are we were first disappointed when we saw these results. These are neurofilament light. You see, it's very messy. Most players do not change in their neurofilament light level. The diagnosis of concussion is liberal in Sweden. If you hit the head and feel a little bit bad afterwards, you're taking off and you get the concussion diagnosis. So that's how it works. But yeah, so first I was a little bit disappointed when I saw this, because I thought we would have a much cleaner biomarker pattern. But now I start to think that this might make sense. So, because the discussion around concussion and the long-term consequences has a little bit been, well, people have started to ask, where are all these people with chronic symptoms? Well, if concussion is that dangerous, why aren't there more people who have problems? But I must say that I think these types of hits to the head, most of them might not injure axons that much. It might be that you feel a little bit bad, but well, nothing happened. But then in some people, you get an injury type that really injures the axons. And that's probably rotational injury. And we have tried to look at all these games are videotaped, not videotaped, recorded. But it's very hard. We just have 2 cameras. And when you look at it's very hard to tell what. And we have asked engineers to see if they could do some kind of modeling and see what types of forces might have been impacted on them. But it's clear that these who take off, at least, they are the ones who often have longer symptoms. So there is a correlation with one of the one of the symptom scores that is used. So the higher the score, the higher the serum neurofilament light level. There is something here that indicates that some types of hits to the head will injure the brain more. And perhaps this is a potential use of the biomarker in this context. If there is a hit to the head, you don't play. There's no need of any acute biomarker testing. But then when you are visiting the medical team 6 to 10 days after the concussion, a blood test could tell you if you are one of those or if it looks like this, and you might be fine to return. And now this is not done, of course, but now neurodegeneration. Here we have worked a lot with the London team. Natalie is here, and Philip West did his thesis on this. So we looked at mutation carriers for familial Alzheimer's disease. Red are mutation carriers, and black are non-mutation carriers. And here you can see mutation carrier who is way before expected year at onset. Here, stable, most are stable here, but you see around 10 years before expected year at onset, individuals shoot off in their neurofilament light concentration. And I think this is very interesting. Now Philip has collected even more time points here, and it looks like there is some kind of plateau effect here. Matthias Jucker's team have also done the whole Diane, and it looks very similar. It looks really similar. So I think this is quite real. And here again, one thinks a little bit about the clinical perspective. The day we have treatments, perhaps it would be, one could find out if I had a mutation, I would know about it and go and check my neurofilament light blood levels every second year or something like that. And when I start to increase, a therapy is initiated and you're brought down to your baseline neurofilament light concentration and maintain there with an effective treatment. And if you start to increase again, one could change the treatment, increase the dosing or something like that. Hopefully this is what we will see in the future. The time points, after you have had an acute event, when should you sample the patient? This is less relevant for the neurodegenerative diseases, but for the acute diseases, I think it's relevant. So here we did a study with Australian researchers, Liz Everett, where we looked at plasma neurofilament light concentrations during surgery. These are hours after initiation of surgery and anesthesia. And we see an increase in most people, and then it goes down. For tau, there is a very strong increase in plasma tau levels during anesthesia. It's not related to surgery type in this study, but a strong increase, and then it goes down. So tau appears to be a rapidly changing marker in blood, whereas neurofilament light appears to be much slower. And it would have been very interesting in this study to have later time points, but we didn't. In this study we had. So here we have tau % increase in cardiac surgery, where you were either randomized to do on or off pump surgery. Off pump surgery, then you will have no pulsatile blood flow. On pump will be no pulsatile, and off pump will of course be the regular pulsatile blood flow. So here we have tau changes. These are on pump people, and these are off pump. So big difference in tau levels depending on. And the anesthesiologist used, I didn't know about that, but pump head they talk about after surgery, cardiac surgery. It sounds almost unethical, but. And here you see the neurofilament light. This is day 7, and there you see the difference. So
the length of surgery, the length of anesthesia in the previous study?
Almost the same, yes. You mean in this study?
Yes.
Yeah, so 3 to 4 hours. 3 to 4 hours were most surgery types, yeah. So there was no clear, and we couldn't address that really. It was quite standardized. Here it's also around 3 hours, 4 hours. But you see that if you're interested in neurofilament light in acute conditions, one should really make sure to capture later time points also. Here is yet another study which we did with the oncology department in Gothenburg. So here we looked at irradiation against head and neck cancer. And here is pre-induction baseline sample at 3 months. And you see neurofilament light goes up. And these are cases who just received irradiation. For head and neck cancer, there will always be a little bit of a spillover of the therapeutic effect on healthy brain tissue. And that can be calculated. So the people giving the irradiation therapy can calculate this. And D2% is the 2% of the brain receiving the highest dose. So here we have increasing doses of accidental brain irradiation, one could call it, or side effect. I mean, it was not meant to. It was meant to kill the head and neck cancer outside the brain. So here are low doses. These are this is baseline, and then weeks following sampling here. So weeks, nothing happens with neurofilament light. Nothing happens. Here the dose is higher to the brain. You see an increase, it goes up a little bit. This could also represent repetitive injuries as you give these irradiation doses repetitively. And here you see that this really takes off in the higher doses. So we can, by this stating that repetitive injuries, 2, 3, 4 weeks, then eventually will lead to an accumulation of neurofilament light in the blood with increased levels. But not much rapid changes. So it takes a few weeks before this becomes clear. Here is also, Jens will show more, this is also from our neurology department before and after natalizumab with serum neurofilament light going down, and then people are maintained. This was also, we did the analysis after the samples had been collected. These individuals got natalizumab, started off low and actually increased, and they had side effects, and they didn't have any benefit of the drug. So this decision to stop was not taken on the biomarker, but it was a bit curious that all of those stopped on the treatment. And then we have addressed in one single study, we have to do more on this. What does it look like in peripheral neuropathies? So this is a study on Charcot-Marie Tooth, together with Mary Riley's team, controls and patients, and then different severity grades here. So there is a slight increase, but not as clearly as in, for example, multiple sclerosis or injury. There's a big overlap. But at least there is a contribution of peripheral nerves, I guess, to neurofilament light concentrations in blood, in spite of the fact that we see good correlations between CSF and plasma neurofilament light. We have not done much work on the other neurofilaments, but we have tried to work a little bit with NFH. So these are stroke data. Here we see CSF neurofilament light, control individuals, day 0 to 1, normal levels, so it takes a while for the levels to go up. Then day 2 to 3, 7 to 9, 3 weeks, and 3 to 5 months. It looks like this for neurofilament light. We have never been able to make a really good NFH assay. So we are struggling with this still, and often we see more variation and less fold changes. But it might be that the assay is not good enough, so we will continue to work with this. Here are the serum neurofilament light data looking very similar to the CSF levels, and the serum neurofilament heavy data are more variable, but we see the similar pattern, so it could be the assay that is not good enough. So to conclude this biomarker discussion, neurofilament light can be measured in CSF, serum, and plasma. They yield very similar results, but neurofilament light levels in blood are a little bit different in different studies, but 1 to 4% of CSF levels, one could say, roughly. It's a promising biomarker for neurodegeneration, neural injury. I forgot to bring in a slide on that, but I had one slide where I showed neurofilament light changes in most neurodegenerative diseases, including ALS and frontotemporal dementia and Alzheimer's. And it's really up in all neurodegenerative diseases, except one, and that is Parkinson. Pure Parkinson has we have no good injury marker there at all in the fluids. Atypical Parkinsonian disorders are elevated like the other neurodegenerative diseases. So it might reflect the amount of neurodegeneration going on. It's a dynamic marker in brain injury. It's a dynamic marker and can detect biochemical treatment effects. NFH assays need more work, at least the ones we have tested and tried to develop. But I feel almost like a drug that doesn't reduce neurofilament light doesn't do the trick. That's of course, now you have to remember that I work in a lab. So, and it might be that there is. But I think that could be a very useful marker in our treatment trials. Not at least thinking about all the results in multiple sclerosis that we will hear more about later. This is the team in Gothenburg and London. Thank you.
Henrik, that was great. Let me just start with questions, but I'll begin scanning the audience to see who might offer questions next. But let me first ask, I was particularly intrigued, I was intrigued by many things, but going back to your FAD data, in addition to the rises that you start seeing about year minus 10, what was very striking is there would seem to be a group of people who were within that proximity, but who were actually, they were up, but then they were also going down. And do you have any sense of whether there are dynamic changes in these chronic elevations that might reflect sort of greater vulnerability to minor traumas of everyday life, or something else?
That's a very important point, and also that is one limitation when interpreting neurofilament light, because it will, I mean, if someone has had a minor stroke or something, I mean, a concussion or something else, of course the neurofilament light will change. So it will be, clinically, it will be very important to try to tell why a person is changing in neurofilament light levels, and not just state that this is initiation of neurodegeneration. So that will be the challenge with this biomarker, to ascertain that the biomarker change reflect what you reflect the disease you think it might reflect. It will change in response to any CNS injury or perhaps comorbidity, and so these are, so most go up like this, but this one, of course, one would like to. I hope that Phil managed to collect additional samples in this individual. We don't know. We will know soon. But these are individuals. There are also resilient people in Matthias Jucker's data, much larger data set. So there are people who, so neurofilament light on a group level could be used also to detect resilience factors, I think. You have a mutation, but you don't enter the neurodegenerative phase as early as expected. What is different with this person as compared to other persons? I think if actually perhaps one could find some genes by this approach if we do large studies. But the short answer to your question is that we do not know why it looks like this on some individuals, but most take off like this, and we think it is due to the mutation.
Well, let me ask, so the related question is talking about, given that there may be dynamics in this, what I'll call mid-elevation range, where it's 2-3 times upper limit of normal or something like that. Is it possible that actually reflects clearance differences? So, for example, have you looked at whether there's a circadian cycling to these? Whether, for example, they're up in the morning and down in the night?
We have not, but that's, one should do that on all these markers, I think, including tau, more carefully. For a lot of work has been done on beta amyloid in this regard. I would, if I would, yes, I wouldn't think so because of the slow dynamics. So I think that those will, I think there will be, I think we won't see a circadian rhythm, but we have to do the experiment to test that, actually. And we haven't done it.
No, thank you. I think you actually addressed the question I had, and I'm always very impressed with the variability, and certainly there's these, it's a dynamic measure, but we also see that variability in the controls too. Yeah. So I think that's really a need to sort of understand, those dynamics in these healthy individuals, and to find the actual controls of the
normal
cells.
aging, stability. Yeah, exactly. A large population of healthy people who age, and also people who get different age-related diseases. That's ongoing actually with some population-based studies where people are followed over time. That will be very important, I agree, and especially it's easy to make reference limits, but then decision points and what is a relevant increase, and that needs a lot of work.
Henrik, I have a brief question on that chart here. Is there gene dose effect for homozygous versus heterozygous in terms of the slope?
I don't know, actually. There are probably mutation differences between different mutations, but that's also something, I hope that might be addressed in Matthias Jucker's larger study. Do you have any comment on this?
These are all individuals with autosomal dominant familial Alzheimer's disease, so APP and preselin 1 mutations. So there's the carriers and the non-carriers, and they're fully penetrant mutations. But I think it would be really interesting to look at their ApoE genotypes, which we know does have a gene dosage effect, and see if, even within people with the same mutation, if that's having an effect.
Clinically, it has a dose effect.
Yes.
Yeah. So in order to bolster the biomarker case, it would be nice to see the same being reflected in the neurofilament light level slope over time. Yeah. Can I ask a second question? You mentioned about the half-life time, 2 to 3 weeks in CSF and 10 hours in blood circulation.
Of tau.
Of tau, yes.
Neurofilament light has much longer.
Yeah.
Much longer.
Can you comment on that?
Yeah, it's the thing we are trying to figure out now is tau is if you spike tau into a plasma sample and put it at the bed, you will lose it. So we are actually looking for if there is an enzymatic degradation going on, really cutting tau between the antibodies of the total tau assay, and they are pretty close to each other. So it must be somewhere there. Whereas neurofilament light, we see no such effects, but then again, we have an assay that measures the rod domain. And it is probably a central small fragment, potentially in some type of aggregator, or because we can also do homogeneous ELISAs and actually take the same capture and detector antibody and get a signal. So actually we are not sure exactly what we are measuring when we are doing these studies, except that it's neurofilament light and probably a fragment.
So I thought the data on the ice hockey players was interesting, that you didn't see a real signal with the neurofilament light in serum. And so my question is for these more minor injuries, is it more important to look at CSF? Do you think you would see a better signal in CSF?
I actually think that we see a signal. So we see a clear signal in a subset, and I think these are the people who have experienced a hit to the head that shook the brain in a way that axons were injured. And I actually think, but I do not dare to, I mean, we don't know this, so we have to continue to work. But I think that these could have been hits to the head that perhaps did not impact the brain that much, at least not from a structural perspective. But that is a very, again, it's a lab physician statement over-interpreting results. But it still, to me, it makes sense that because otherwise we would have seen a much worse epidemic among all the professional ice hockey players. So some are a little bit unlucky, and perhaps you have 1 or 2 hits like this, and then it's. But then perhaps if you have 3 or 4 or 5, then you might enter something that is not good at all.
It's on the ice hockey and the concussion again. Do you think there is any correlation between the degree of the hit? Because I think what's been seen clinically in concussion is that often it's not related to necessarily the violence of the injury. There's a subset of people who, even with minor concussions, do very badly clinically. Yeah. And I wonder, is there something pathophysiologically? I know there's that relationship with migraine pathophysiologically potentially, or are we seeing with that NFL rise not necessarily a bad hit, but a vulnerable population who, even with a reasonably minor hit, are going to have a greater degree of injury than would be expected?
Yep. We have examined ice hockey players with post-concussion syndrome. It's a little bit the clinical criteria are not very good for that, but they are on a group level higher. They actually came in for a CSF sampling. So we had around 25 ice hockey players who do not feel well. It's hard to tell if it's what it's related to, and there are other potential confounding factors. And then compared to ice hockey players of similar age who have had concussions but are okay, and they are on group level higher in neurofilament light, but not all. So it is a heterogeneous population, but a subset have biomarker evidence of ongoing axonal injury. Many of those players have not played for 2-3 years, but are still higher in neurofilament light. Of course, they could have another disease also, but they are between 30 and 45, so they shouldn't have, at least not that many.
So it's related to this also ice hockey study. But it's also a more general question. Do we have actually very nice brain imaging, or can we see that in the future that could really assess whether these different discrepancies account for real axonal damage?
Yeah, I think definitely that, better MRI, and also there will be a lot of things going on there. We had a conference in Stockholm with also imaging people discussing these matters. And with standard, CT and standard MRI in the emergency care room, it's very hard to say anything except excluding a bleeding more. But I mean, for axonal structures and white matter changes, and I mean, there is enormous technological development. But here we have only done the fluids, and we would love to do more advanced imaging studies in parallel with the fluids.
And a short question for the fluids. You said that you have difficulties in performing NFH-related test. So is it a problem of the antibody or the composition of the blood? Meaning, could you perform like EM study, classical studies, to just see that the material is there to start with and see if it's a technical development rather than the presence or...
Yeah, I don't know, but I mean, that's a very good point. And we haven't done that much work on NFH yet, but that's something we could discuss, actually.
To follow up on this point, what kind of antibodies are you using for NFL or NFH? Are they monoclonal or polyclonal?
Yeah, monoclonal. So for most of the NFL data are the human diagnostics monoclonals. But now we have made our own monoclonals that sort of work very similarly to the human diagnostics antibodies also. So monoclonals. For NFH, we have also done screen for monoclonals.
So for the NFH, it could be a problem if it's phosphodependent.
Yeah, yeah.
So maybe a pretreatment with the kinase.
Yeah, exactly. Yeah, that's a good point. That's a good point. Yeah, perhaps. We could both phosphorylate and phosphorylate and see what happens. That's a good, very good point. Yeah, exactly. That's a good point. Yes.
Oh, sorry. You mentioned how you wanted to potentially see the effects of treatments on NFL in clinical trials. And in the familial Alzheimer's disease, you mentioned it might be used as a screening tool. I'd be curious your thoughts on how it could be used as a screening tool potentially for recruitment into clinical trials, taking into account the age effect and also the fact that it's more dynamic, like you've presented today. So you have the amyloid positivity and negativity, for example, in Alzheimer's disease for BACE inhibitors. What do we have to do to start thinking of NFL as an inclusion criteria, let's say, in a clinical trial?
I think it all depends on the mechanism of action of the drug also, because one could think of drugs that are aimed at detoxifying existing amyloid pathology. And then, of course, one would like to know that the patient has amyloid. One would like to know that the patient is reacting to the amyloid. And then neurofilament light could be part of the inclusion criterion. But it might be that one would need to include on the basis of 2 samples and actually look at the slope. But of course, this is not an easy study design. But that's one potential way of using it. If one would think positively about the BACE1 inhibitors and try to just forget about what has happened recently, if the Icelandic mutation thing is true, that if you have a 30% reduction in your beta amyloid production, you are more Icelandic mutation-like, and you age without getting the cascade taking off. If one would then be in a family with Alzheimer's and you start in a clinical trial when you're 35, 45, you would like to know that this individual is amyloid negative, and you reduce the BACE1 activity with 30%, and then you use the biomarker to tell that amyloid doesn't show up 10 years later or something like that. And then I would also expect NFL to stay normal in relation to the expected age-related increase. But I see that it's not easy to, it's a little bit challenging to use it. But in a symptomatic phase of any disease like Huntington or frontotemporal dementia, if you do a down-regulation of the pathogenic gene, and so then of course you would expect to start off high and then go down. That's the simple scenario, I think. And if you start with the patient population with amyloid and increased neurofilament light, you would like to affect the amyloid pathology, and then if that does something good to the neurons, one would expect a reduction. But perhaps it would still be, if it was a longer study, if it was a 2-year study, one would still have to consider the expected 2-year increase in neurofilament light. And there is more and more data on this. It alludes to your question also that we need to understand this better. What is the normal age-related change in neurofilament light?
So your last comment is exactly on point I wanted to ask about. There are so many vaccine trials and phase 3 trials of various treatments. I would think it would be of interest to compare them across the different treatments, as well as just to look at the treatment, any given treatment, in terms of, have you done that?
No, so that is definitely something we would like to do. And I mean, those trials that have been stopped, it should be doable.
And would they be willing to give you access to?
Yeah, we have talked a little bit, but we have not yet done it. But I think it would be very interesting. Very interesting. Now I see Andre also. I forgot to mention, I should have brought slides on Down. So we could, if you want to discuss Down and neurofilament light data, Andre is in the back of the room. It looks very similar to the familial AD data that I showed.
Sorry, can I just ask a practical question? In fact, we have got samples for about 200 patients having intervention. So we have, collected the blood and kept it in -80. Is it actually practically feasible to do the NFL on those samples?
Absolutely, yes. And the NFL is such a stable mark, you can actually mistreat the sample quite a bit.
Okay, thank you.
And still get a good... virtually nothing happens to that protein. Again, the way we measure it with the mid-domain, raw domain antibodies close to each other. A lot of things are probably going on in the ends of the protein, that I'm sure of.
Very nice talk. The Huntington's disease trials that are ongoing with intrathecal administration of antisense, would it be useful in those trials to monitor?
Yes, absolutely. And there I think there are things going on with such measurements. So that's a typical. Again, the SMA data indicate that it should work. I think and Huntington definitely has increased neurofilament light concentrations. The higher the increase, the more rapid progression. So it seems that it's really a disease intensity marker there also.
Well, I think we could go on. I mean, this is such an important area. This is the core of, in some respects, the core of the problem we're trying to work towards. Let's stick with this theme. And Henrik, would you like to introduce Jens?
Yes,
next?
Absolutely. So there we have Jens. So Jens and I have collaborated a lot, and Jens has done a lot of work on neurofilaments with a particular focus on multiple sclerosis, but also other inflammatory diseases. Please, Jens.
And while Jens is coming up, I should have said, and let's all thank Henrik for an incredibly stimulating.
So thanks a lot for inviting me. As Henrik mentioned, we had a deal before, and originally the methodological considerations were my main subjects, and I have now built in bits and pieces of methodological issues about assays in MS results, basically. This, I think I can jump. There has been a lot of CSF data over decades in various neurodegenerative inflammatory diseases. The assays were considerably not sensitive enough to reliably measure the approximately 50-fold lower levels in blood samples of NFL here in this case. This assay had a relatively short half-life because I met this guy, Henrik Zetterberg, in London when I was here 2 years.
in 2012. He was telling about the Simoa technology at a seminar at UCL. And I saw this assay already going down when he gave his talk about Simoa, which is much more sensitive for blood measurements. And this has also turned out to be the case. So this was an electrochemiluminescence assay using the same monoclonal antibodies as 99% of people are using to measure NFL nowadays. Bovine NFL as a calibrator, and this was a non-MS study, but we saw higher levels in serum and in CSF, and also saw high correlation, but we had a problem with this assay in the healthy controls and control patients in serum. So, and this is what Henrik explained to you. This is this bead-based Simoa technology, quite a huge machine. I think Gothenburg has 3 meanwhile, or 4. Basel has 2 of them. So we are quite busy measuring thousands of samples, actually. And the reaction works on these beads. It's like a normal ELISA system. And the trick is really these 50 femtoliter small reaction chambers that produce a high signal-to-noise signal and have a very low dead volume, and that lead to this more digital outread. This is also a magnification of these beads, and each sample is measured, I think, in 160,000 of these small wells. So together with the colleagues in Gothenburg, with Kai and Henrik, in I think 2013, we said let's compare the mesoscale assay, the ELISA and the Simoa assay, all using same antibodies, same calibrators, and we selected pairs of serum samples based on mesoscale measurements. So we chose 9 high serum NFL samples, 8 medium. Pairs was CSF and serum, and 7 low. And also in those days, the field was quite convinced that there is no blood biomarker for MS. So the field was quite convinced that NFL, neurofilaments in blood samples would not make sense in MS. And we just set off with 9 pairs from MS that had not been measured before to stick them in these 3 systems. And we had this more practical definition of analytical sensitivity based on the lowest calibrator fulfilling certain criteria. The ELISA, this was the lowest calibrator of the ELISA system. I mean, maybe there is also some potential in more sophisticated ELISAs if one uses lower calibrators, other technologies. ECL 1578 0.62 with the Simoa assay. What was interesting that the level samples below sensitivity were similar between the ELISA and the mesoscale assay, but this was driven mainly by a few very high NFL concentrations in ELISA. I guess to analytical people, quite known phenomenon that if you don't use any heterophilic blockers against anti-animal antibodies in humans, then you can have false positive results. And interestingly, this well-known and broadly used ELISA doesn't really use these reagents. So samples which were very low had very high levels in the ELISA system. And this obviously the Uman Diagnostics ELISA assay is critical to use in serum or plasma samples. When we did correlations between serum and CSF, we saw that levels were highly correlated for the Simoa system and the problems with the sensitivity in the other assays. When we looked at correlations between different platforms in CSF, it didn't really matter if we use ELISA or mesoscale or Simoa, they were all highly correlated, extremely highly correlated, 1.0 correlation coefficient here. And in serum, best results with the Simoa and the ECL assay with the limitation of the sensitivity of the ECL assay. And when we looked at the MS samples, we had a significant difference in MS versus the low samples for the Simoa assay and no significance versus the low samples, which could be somewhat controls, healthy controls for the other systems. So this was the end of the mesoscale assay, more or less. And actually, Kai and Henrik helped us also to develop an NFL assay at the University Hospital in Basel. And since then, we are using this so-called, by the company Quanterix homebrew assay. We have been using this for many different studies, and we also had access to different clinical trial samples which were sitting in the freezers, just like you mentioned. So we did approach companies, and this has been really a quite interesting story over the last 3 years. This I already showed. I mean, this transition to blood-based measurements has really opened ways to less invasive testing and making longitudinal studies feasible. And in MS, really, we have, the luxury problem of having approximately 15 different drugs at hand for our relapsing remitting MS patients. And patients sit in front of us, and we do MRIs, and we speak with patients, and we don't really know if the drug is doing its job in these patients in many instances. So quantifying MS, doing individual treatment decisions is heavily based on examining the patient, speaking to the patient, and on MRIs, which often also lack the spinal cord imaging, for example. So in relapsing MS, we are eager to have better biomarkers to stratify these different drugs that are available. And of course, we are extremely bad in prognostic estimations, and this is a huge unmet need in the MS field. And so this was also basic motivation why I started trying to measure neuroaxonal injury measures about 8 to 10 years ago. And I've structured the results in MS in different chapters, and we will go through one by one. So first step, we looked at blood NFL as a measure of current disease activity in MS. We also looked at paired CSF and serum samples, and we saw this high correlation between serum and CSF, which also Henrik already mentioned. And we saw that patients with MS with a higher brain T2 lesion load had higher NFL levels in the blood. And this was also true for gadolinium-enhancing lesions. Patients with 3 gadolinium-enhancing lesions had significantly higher levels than without gadolinium-enhancing. We look at the group comparisons. We saw that relapsing and CIS patients, and especially progressive MS patients, after age correction, and I will get back to that, have significantly higher NFL levels. And more disabled patients here on a scale up to 10 also have higher NFL levels in the serum here. In the multivariate model, age is an independent predictor. EDSS step, relapse, patients having experienced a relapse within the last 2 months had a 10 picogram higher median NFL levels. Interestingly, also in this observational study, where one could assume that more severely affected patients are receiving the most effective treatments, patients that were treated had approximately 11 picograms lower NFL in the blood, which was the first indication that treatment, even in the real-world setting, could potentially have an effect on these measurements. Now, healthy controls. This is a plot of 400 healthy control values. NFL on the y-axis, age on the x-axis. And you see that levels increase with aging. The spread also gets larger with aging. We have approached this by generating percentiles. This is the 99th percentile. This is the 80th percentile. So 20% of healthy controls lay above this line, 80% below. And then we investigated several sets of samples comparing patients above versus below these percentiles. And just a few words about this. It's hard to believe, but it could be true that this represents aging, right? Age-related neuroaxonal injury, which is difficult and we don't really know. And a few thoughts regarding this. This is Henrik mentioned it, the albumin quotient as a measure of blood-brain barrier function. And we do know that children have an extremely high albumin quotient, so a severely injured blood-brain barrier, which normalizes up to the age of 5 and then slowly increasing over the years. So could this just be spillover from a higher CSF concentration, CSF to blood? Then Henrik's group had this paper here, where they looked in patients with increased albumin quotient, normal albumin quotient, and correlated it with NFL. I think they were quite happy that they didn't see a significant association. CSF and serum highly correlated. when we measured about 300 children, healthy children, we see this decrease of NFL in the first 5 years. This is unpublished in collaborations with colleagues from Göttingen. Again, making me, of course, somehow think about this curve here, right? And I don't have a solution. I just want to present it and discuss it with you. I mean, what is, though, important is that in CSF, we also see the age-related increase. So it seems not just to be a spillover in blood, in severely disabled blood-brain barrier, because if that was the case, we wouldn't see this age-related increase in CSF, also in the CSF. So I think it may be a combination of these two mechanisms, but this regarding dynamics is also interesting and important to look closer, I think. So in MS, looking at disease activity, NFL is really, it seems to be a first-in-class blood-based MS disease activity marker. So we were quite encouraged by these findings, and in the next step, we looked at treatment response, more at treatment response, and this is the data that you saw before from the Swiss MS cohort study, where we had samples before switching to treatment, to a different treatment. These were samples before, 6 months after, and 18 months after a switch. These are the healthy controls, and we do in Switzerland, fingolimod is first-line therapy, and it was interesting to see that doctors switching patients to high-activity treatments, patients had higher levels before switching than patients switching to first-line treatments. After treatment switch, levels went down to some extent and with some heterogeneity and with some overlap and with all the difficulty of an open label design, which could imply that the injectable DMTs are the most effective treatments in MS, which is not really the case. And similar data by Henrik's group, together with the colleagues from Sweden, when you switch treatments, initiate, escalate treatment, levels go down. Also data by Fredrik Pihl and Thomas Olsson, they also see a significant decrease of NFL when starting fingolimod treatment. So with regards to treatment response, blood NFL is a marker of therapy response. The challenge, as people also here pointed out, is to establish cutoffs for individual decision making, and this is something towards the end of my presentation that I think would be nice to discuss also here about this, what we still lack to do to go in that direction. Now, looking at prognosis, MS, it's difficult to predict MS. I don't know if it's possible to predict MS, and it was not for me necessary really to find anything here. But anyway, it's of course nice if something you measure in blood predicts the next year or predicts the next 5 years. We looked here at brain volume change over 2 and 5 years. Brain volume change in MS is the key outread for neuroaxonal injury. It's the best it's facilitating said it's the measure that is used on a relatively broad group level. And when we compared patients, this is 2-year brain volume change, this is 5-year brain volume change, and these are these percentiles. So when NFL levels increase and when you compare the patients above this percentile versus below the percentile, we do see that patients have clearly more brain volume loss over the next 2 years. This is the 99th percentile. So these were only 23 patients in that group, 174. But this dose effect is somehow also special to see with increasing NFL levels, brain atrophy increases also over 5 years. And in the multivariate models, NFL was the strongest independent predictor of brain volume loss over the next 2 and 5 years, correcting for normalized brain volume, T2 lesion volume, contrast-enhancing lesions, EDSS with some slight effect. And the same was true for spinal cord volume. We could measure a small block of spinal cord volume in the brain MRI scans in this study. And this was also true for 2 and 5 years with clearly more additional volume loss over 5 years in the spinal cord if patients had high NFL levels at baseline. Last chapter, we tried to now look what can we do with this serum NFL in patients that are on established treatment. So the question here was, can our first blood sample 3 to 24 months after treatment start predict future disease activity? So identify suboptimal treatment response in a real-world setting. The Swiss MS cohort study follows about 1,200 patients every 6 or every 12 months with a biosample and an MRI imaging. So Gavin Giovannoni, who I spent 2 years with, had this editorial, and I don't know, so maybe we need to discuss that. And the last part, we are doing a quite large multi-site analytical validation of the commercially available, from Quanterix available Simoa NF-Lite assay. 17 centers are currently measuring standardized materials, and I can present you preliminary results from 6 of these centers. Also, Gothenburg, Basel, and also Sanofi is participating in this. Quanterix is also actually not on this list. They are also measuring. So thank you for your attention. I'm happy to answer any questions.
Great. Well, that was, I think you've really raised some critical questions at the end. Let me just go back to something that we touched on earlier in the day, and that was differentiating peripheral versus central effects, and particularly in multi-trauma patients, for example. Perhaps could you and Henrik and maybe anyone else speak about the challenges to, for example, getting an assay peripherin or?
You mean getting an assay that could dissect that?
Yeah.
Yeah. It's somehow reaching the stars. It would be nice to have. Peripherin seems to be more difficult than one thinks, and I think you're working on alpha-internexin potentially. I cannot judge that. It's important to know that this is a potential confounder, of course. Yeah, but this is so far, to my knowledge, not completely solved. Don't know if you have a comment to that.
I think it's. Oops. I think it's what you said. It's we have to have. I think alpha-internexin is definitely one assay we should try to develop, and perhaps peripherin we have tried, but it's been really hard. It's we don't understand why. We might come and ask you guys for advice. So I would really favor to have all these filaments together and then do studies in parallel in peripheral and central neuropathies and compare.
For MS, have you ever tried myelin basic protein?
We are working on that. We are working on that to quantify demyelination and also potential other sphingomyelins, other components like that. It would be quite obvious to try to do that. There are ELISA data around for MBP and CSF, and Simoa platform is open for such developments, right? Have you MBP? Thank God Henrik's group is basically interested in dementias and not so much on MS. Otherwise, and we keep it like this.
So, Henrik mentioned earlier a very intriguing observation about marathon runners. Did I hear this right? It sounded very scary that the blood levels of NFL were very highly up.
Component T, I just alluded to that. It was not.
So Frederick looked at it. Okay. Frederick Peel did 2 lumbar punctures before and after a marathon, and there was no increase. It was normal after the marathon.
Okay. Well, I guess without having that as an implication, then what about a muscle disease, a pure muscle disease, where you might have severe atrophy? Would that give you some kind of handle on peripheral versus central, or is that still messy?
Well, the beauty is the high specificity of the neurofilaments, right?
Yes, but I'm thinking that the peripheral nerve atrophy that might come along with the muscle atrophy or
okay, directly, yeah. Maybe. Look in ALS, CK, and things like that, but I don't know. Yeah.
But I see what you mean, to do something which is, the muscle is decaying, and then you have the peripheral, perhaps dying back of axons.
Right, because, I mean, even in ALS, I mean, you're talking about, there really hasn't been very much tension, as I understand it, between a pure peripheral condition and many of the things you're studying, which are more central.
Yeah.
So I am very impressed and intrigued by the predictive value of neurofilaments and disease progression upon treatment. So could you tell us what are your guess about the possible mechanism? Is does that imply that there is a threshold in neuronal damages that can make impair recovery, or is it like a genetic susceptibility?
It seems like individuals have different levels of Basel NFL neurodegeneration that over time lead to a worse disease course if they are kept up, right? Something like that. I have to say I wouldn't have expected such a predictive value over years, because I thought NFL, the neurofilaments are relative dynamic disease activity. So I have no clue about the mechanism except the obvious one, that more neurodegeneration uses up reserve capacities and people get symptomatic and lose more brain volume by that. Yeah.
And this also implies that in the normal situation, because here we are really below what you showed for in acute damage, so it means that there is a lot very subtle differences that matters in the within the normal range, and this is also
normal
something
range is a difficult subject and a difficult term at the moment, because there are different assays around. Some assays produce higher levels, some assays produce lower levels. So normal range, what do you mean by normal range?
The one that do not respond like massively to the damage. You know?
Yeah, I mean, this is a subacute, very chronic disease, and there we just have not these peaks, like in boxers, yeah. It seems like it matters also for the prospective disease course. I cannot tell you the mechanism really. That's a problem we have.
Yeah, actually, I'll just add a comment here, and you might reflect, you might add to, and then we'll go around. We have some questions around the audience. But one of the things that one finds in MS or in other autoimmune diseases, for example, IBD, is that an acute flare leads to a chronic inflammatory insult. So if you look post-mortem on neuropathology, while there are inactive lesions that really seem to be cleared of any inflammatory activity, they're actually not that common. And very common are lesions that have had prior acute activity that retain an active rim driven by macrophage microglial activity eating away continuously. So, you know, the way, I mean, and this is for you to reflect on, I mean, what I'm wondering about is you know, whether what you're producing a measure of inflammatory trait and reflecting past history in part. People who have had worse disease have more lesions, more acute lesions, therefore also have more chronic inflammatory activity, which isn't really being so well touched by our current drugs.
Yeah. I mean, I think what we have really in MS is a lot of subclinical disease activity that we don't capture. So we know that in MRI we have many more flares than we see clinically, right? So there's a lot of subclinical disease activity going on in MS. Neurofilament is a common an end product of any mechanisms leading to injury. We cannot really dissect if this is inflammatory or not. It does seem to be independent also of inflammation measured by our means. At the moment, there is some data analysis ongoing showing independency of acute inflammation also. So the inflammation in MS may need to be redefined based on which measures you have, right? It's a difficult, very difficult subject. So this is not the role of NFL dissecting these mechanisms, but in MS it's very important to have a measure of the substrate of disability potentially in the blood, right? That's something important there.
Thank you for your work in advancing this field. I think at the beginning of the talk you mentioned that we need to establish cutoffs for individual treatment decisions, and we should have some discussion on that. And I think I'll bite on that and ask you, are we ready to make decisions or individual patient decisions with higher or lower NFL levels?
I think we've never been closer to something like that, like today here in London, I have to say. Yeah? And it's already amazing that we can think about this, and it's maybe even realistic. So I think it's not completely impossible. I think ideally we would need to decide an assay format. We would need to agree on the assay format. And we need to generate large normative data sets to define percentiles and reference ranges. And we may need some prospective trials, and I think we may need to still increase face validity of this biomarker in MS despite all the data that is there. So these are things I think that should go now in parallel, and there's a large room for improvement in MS, obviously. So it's not completely impossible, I think.
Just a short comment related to this. Another way of doing this would actually be to create a set of reference materials, because then we don't have to decide on a specific assay. That is always hard to do. It's good that there are many companies competing, but if we have a common reference material with which one can the kit vendors can calibrate their calibrators, sort of. The prerequisite is that we do commutability studies on the different assays on different samples, and then look at the correlations, because if we see that the different assays correlate well, then they can be standardized to each other using this type of certified reference materials that we have developed, for example, for CSFA beta 42 now, and that is actually also a very common clinical chemistry way of working, because then we don't have to discuss which assay to pick. Instead, we could create something as researchers, as tools that then could, if they are certified by, for example, JCTLM, the Joint Committee for Traceability in Laboratory Medicine, then the companies have to use those reference materials to set their calibrators. And I think that could be we should really do that now, I think.
Just a couple of comments. The first is you mentioned the correlation with age and how that's going to have to be corrected for, but there are an equal number of manuscripts and publications which suggest no correlation with age and neurofilament. My second point is I wonder, going on from what Henrik just said, whether we can learn from other disease areas such as oncology and how they've developed their biomarkers as a way of working out the practical steps that are involved in actually introducing this.
I think there are no data on healthy controls and age not correlating. I think if you use healthy controls, you always see an association with age. In MS and other more traumatic or severe diseases, the age effect is covered by the pathology sometimes. Then you don't see that anymore. I think that's a reason why in patients the age association is a bit fluctuating, but in the healthy controls I think this is a very consistent finding of in CSF and blood that you see this age association. Or are you aware of publications in controls where you don't see an age association?
I think you have a publication in MS Journal which suggested, but it was in the context of MS patients having the correlation not being clear.
Yeah. I think that's what we see, that if patients have 5 gadolinium lesions, age is not visible anymore, right? And sure, I mean, that's why we are here and discussing, because we should define a way forward, I think, as a group, ideally. And the oncology field has made great progress here. I mean, from practice, I have to say, numbers are increasing from doctors asking for determinations of NFL in blood from their patients. This is important to canalize, ideally, to see what, not to give false advice also is important here.
This more general question, when you don't have a neurodegenerative disease, but you have misconnectivity, for example, like in syndromic autism, you think there will be differences in the secretion of NFL?
We didn't have the chance to look into such samples, for example, in these diseases. Difficult to say if this would lead to. I mean, I think it's not super sensitive sometimes, NFL in blood. I think you need a certain level of damage to find. I think you had a heading study recently, and in the heading people doing headings, it was normal, or? It was CSF or serum?
CSF and serum.
CSF and serum.
First we started to do a CSF study and saw nothing. In the serum study we see a little bit of reflex of heading, but it's very minor as compared to what you're seeing in sclerosis.
I got a very technical question. So I totally agree about the need for standardization across sites, across different labs. For the multi-validation site, I think the results are great, but as you know, all the material was from the same batch, same lot number, everything was very carefully controlled by Quanterix. One issue I have sometimes is that different batch performs slightly differently. So I was wondering if you're planning to sort of work more on that, to just not only reproducibility of results within labs, but also labs using different batches of material can continue to be confident in the results they have.
Yeah, obviously this is a key point to solve if one wants to use this biomarker. One has to say MRI also made it somehow into clinical practice. It's difficult to standardize MRI to the maximum. But yes, it's a key point and it's a difficult point, but maybe some calibrator samples would still be very important, right? General calibrator samples maybe in different levels, and that could maybe be a good way forward instead of trying to convince the world of one single assay.
I could also make a brief comment on that. So it's also very much up to the kit vendors, because now the current versions of the commercially available kits, they are research grade. And that means that the batch release criteria from the companies are not that strict. But if there is an IVD assay, then the release of new kit lots, that has to be bridged very carefully. In the meantime, as a laboratory, you can live with this, as if you actually build up a little bit of if you have internal control samples, that's one thing, but you could also have a set of reference samples that you actually can run together with your new study and have, for example, 20 or 30 samples that you actually run all the time, and then you could, in a scientifically sound way, normalize your runs to each other. But that is sort of the laboratory's way of dealing with the lot-to-lot variation one can see for research grade kits. But in the future, I definitely think that this should be an assay that is released in a way that clinical chemistry labs can buy kit lots that are stable over time reasonably well. And of course, one would still have to use internal control samples and have external quality control samples to check that this is really working also. And the reference material, I think, will be key.
A couple of questions back here, and then we'll go to Jean-Pierre.
So, I mean, using NFL perhaps as a sort of surrogate outcome in clinical trials of treatment, it looks like the MS field is quite way ahead of many other kind of disease areas. And how long do you think it takes before the NFL starts to show a difference in response to a treatment? An effective
How long does it take until the drug works in MS? Big variability there, and we don't really know. because the disease itself also changes without the drug.
But say in a trial, if you had a placebo
in the trial, we saw at 6 months, right? On the group level, we saw a significant effect at 6 months. Depends in progressive MS. We didn't see at 3 months, but we saw something at 6 months. And we didn't have the opportunity to have high-frequency sampling in other trials to really explore that. But we explored NFL as a surrogate for a phase 2 trial in relapsing remitting MS, and there it performs to similar quality as the new T2, new or enlarging T2 lesions, which are currently the main paradigm for such studies. So there, in relapsing remitting MS, NFL at month 6 could be a quite useful outcome for phase 2 studies in relapsing MS. This will be modeled and explored from the Freedoms trial.
Jens, this kind of, over here. Jens, this kind of follows on from Paul's comment, perhaps the last one, is it concerns me a little bit that that you've shown that interventions with therapies that don't affect progressive disease reduce neurofilament levels. So in some senses, is this useful? And then, but not to be too negative, I'm intrigued by the fact, and Paul alluded to it, is this just another marker of inflammation in the cord? When you show brain atrophy and associations, I'm very mindful of the fact that white matter is next to CSF in the cord, but actually buried deeper within the brain, within the sort of the forebrain. Where do you think the signals are really coming from? And are the efficacies really related to the fact that you're just suppressing inflammation in the cord and protecting rather than the brain? So I'm just lots of things there, sorry.
So 3 questions. Yes, seeing an effect on NFL in trials that were negative, right? Informs and expand. I don't know if you have ever done an EDSS. When I did an EDSS in the UK, patients scored 1 to 2 points worse because where I had to walk the patient was uneven. So the EDSS was 1.5 points worse in East London than in Basel. So that may be one of the reasons why these trials may be negative, potentially. And maybe the drugs will work in some patients and in others not. So this could be heterogeneity, and one may be able to define where these drugs may work and others where they may not work. Where does the signal come from? We saw that in spinal cord relapses, levels were higher than in brain relapses. So yes, likely that spinal cord with the large myelinated axons leads to higher NFL liberation, right? May this only be another marker of inflammation? I think time will tell us, and there's a lot of evidence that this is not the case, and we will present some data at the AAN meeting where we try to dissect inflammation from non-inflammation in predicting disease activity, and there it seems like the signal is also there in patients without relapses and in patients without gadolinium-enhancing lesions. If this means this is inflammation or not, and if there's also inflammation despite no relapses and no gadolinium, is a totally different question. What is inflammation in MS, I think, is ill-defined. So this is I cannot really answer your questions, but this is more my thoughts to that.
Do you know if patients with MS develop autoantibodies against neurofilament or other proteins?
Yes, we looked at this a little bit. We looked at it in ALS and we looked at it in MS. I think the assays are quite variable and difficult to determine anti-NFL antibodies. It's not solved completely. We analyzed the data actually that I showed you from the Swiss MS cohort study. We didn't see a lot when we analyzed anti-NFL. We had a project there where we measured it together with the colleagues in London, actually, where we didn't really see a lot of signal in any direction.
SOD could be another one.
It could
interfere, very stable
right?
protein.
Yeah.
Sorry, could you repeat that, Jean-Pierre?
That SOD1 superoxide dismutase is very stable protein. Difficult to degrade, actually, the native form. And once I receive a sample from the blood bank, I just wanted to have a control of immunoglobulin representing, I think, 10,000 people in the normal individuals. And I was surprised there's a lot of antibodies against SOD1. In the normally, we have antibodies against that.
So we didn't see a close correlation between NFL levels and anti-NFL. That's, I mean, a simple analysis there we didn't see, also not with the time shift, which may be the assay or biology, not sure. Conformation, antibodies.
Okay. Well, Jens, thank you. That was. Thank you. Let's all give him a great hand. That was another great hand. Now, I mean, it didn't look like energy was flagging by any means, but let's take a break for about 45 minutes. Luncheon is outside, and you can get some lunch, refresh yourself with a walk along Exhibition Road, and we'll meet back here at 1:15.